Hello there, and welcome to Nocturnal Notions. Tonight, we're stepping away from the bright, busy world and into the quiet clarity of the dark. Close your eyes for a moment, and imagine yourself sitting by a calm lake at twilight. The water is perfectly still, reflecting the fading light. Now, picture a single raindrop touching the surface. Ripples spread outward, gentle and inevitable. But here's what's remarkable: the water doesn't break. It responds. It adapts. It knows, in its own way, that something has happened. This is where our journey begins tonight, at the very edge of damage, in that instant before anything truly breaks. We're going to explore one of nature's most elegant secrets, the way living things know when they've been hurt, and how they begin healing before we even realize anything has happened. The whisper of damage. In every moment of your life, trillions of cells are quietly monitoring themselves. They're listening, not with ears, of course, but with something far more ancient and profound. They're feeling for changes, sensing the subtle shifts in their own structure, waiting for the first whisper of damage. Think about how you might notice a paper cut. There's that sharp moment of awareness, followed by pain, then the sight of blood. But your cells knew about that cut before your brain did. They felt it in the very instant it happened, perhaps even a fraction of a second before the blade completed its path through your skin. This is because cells possess something extraordinary: the ability to feel damage as it occurs. Not after. Not later when inflammation sets in. But now, in the present moment, as forces are being applied to their delicate structures. The ancient watchers. Deep within each of your cells, there are tiny protein structures that act as sentinels. They're called LIM domain proteins, and their job is beautifully simple. They notice when things are being stretched too far. Imagine a suspension bridge. As weight is added, cables stretch and strain. Engineers place sensors on these cables to detect when stress becomes dangerous. Your cells do something remarkably similar, but they've been doing it for billions of years, long before humans ever dreamed of building bridges. These LIM domain proteins sit attached to the cytoskeleton, the internal scaffolding that gives your cells their shape. Under normal conditions, they rest quietly, monitoring the gentle flex and flow of cellular life. But when damage begins, when something pulls too hard, when a membrane starts to tear, when internal structures begin to overstretch, these proteins respond instantly. They don't wait for a signal from somewhere else. They don't need permission. The force itself is the signal. The stretching activates them directly. When a LIM domain protein senses excessive force, it undergoes a shape change. This transformation is like a key turning in a lock, and it triggers a cascade of responses. The protein rushes to the site of damage, recruiting other repair molecules, beginning the healing process before the structure has even fully failed. Across the ages. What's truly beautiful about this system is its antiquity. The same basic mechanism exists in yeast, single-celled organisms that have been on Earth for hundreds of millions of years, and in your own body, with its trillions of specialized cells. From the simplest fungi to the most complex mammals, this force-sensing system has been preserved, refined, and passed down through countless generations. It's one of life's fundamental solutions, so elegant and effective that evolution has kept it, essentially unchanged, across vast stretches of time. When you think about it, you're carrying within you a technology older than trees, older than fish, older than almost anything we can easily imagine. And it's working right now, in this very moment, maintaining you. The calcium signal. But sensing damage is only the first step. What happens next is even more remarkable. When a cell's membrane is damaged, when a tiny hole appears in the delicate barrier that separates the cell's interior from the outside world, something crucial happens. Calcium ions rush in. This calcium influx is not an accident. It's a signal, an ancient alarm that triggers an emergency response that has saved cellular life countless times across the history of our planet. Picture it. The cell membrane, thinner than a soap bubble, has been breached. In the vast ocean of fluid surrounding the cell, calcium ions are abundant. Inside the cell, calcium is normally kept at very low concentrations. The cell works hard to maintain this difference, pumping calcium out constantly. But when a hole appears, this carefully maintained balance is disrupted. Calcium floods in, and its arrival is detected by specialized proteins waiting just beneath the membrane. These proteins spring into action with remarkable speed. They're called annexins, and in some organisms, they can seal membrane lesions within seconds. Seconds. Think about that, the time it takes you to blink, and a cell has already begun repairing damage that would otherwise be fatal. The patch response. The repair mechanism is wonderfully elegant. When calcium enters, it triggers nearby vesicles, tiny bubble-like structures floating in the cellular interior, to fuse with the damaged area. Imagine you're trying to patch a hole in a boat while it's taking on water. You'd grab whatever material you had and press it firmly over the breach, right? Cells do essentially the same thing, but with molecular precision. These vesicles rush to the wound site and merge with the torn membrane, their own membranes seamlessly integrating with the damaged area. It's like liquid bandages flowing together, healing the breach from within. The calcium doesn't just trigger the response, it facilitates it. The presence of calcium helps the vesicle membranes and the cell membrane merge more easily, lowering the energy barrier for fusion. It's a system that uses the problem itself, the influx of calcium through the wound, as part of the solution. The resilience of single cells. Some of the most dramatic demonstrations of cellular repair come from experiments with large, single cells. Scientists have worked with cells so big you can see them with the naked eye, like the egg cells of Xenopus, the African clawed frog. These cells are remarkable. They're preparing to become entire organisms, carrying within them all the machinery needed to divide, specialize, and eventually form every tissue in a frog's body. They're precious, complex, and surprisingly durable. Researchers have deliberately punctured these cells with fine needles, creating wounds that would seem catastrophic. And yet, within moments, the cells reseal. Calcium rushes in, vesicles fuse, the membrane patches itself. Minutes later, the cell returns to normal function, as if nothing had happened. Think about the implications. A single cell, with no brain, no immune system, no outside help, can repair itself. It carries within its own boundaries everything needed for survival and restoration. Amoebas and annexins. Perhaps even more impressive are amoebas, those shape-shifting single-celled organisms you might remember from biology class. They move by extending parts of themselves, flowing through their environment like microscopic blobs of animated jelly. Their lifestyle makes them particularly vulnerable to membrane damage. As they squeeze through tight spaces and engulf food particles, their membranes are constantly being stressed and occasionally torn. But amoebas have mastered repair with extraordinary efficiency. Using anchin proteins, they can seal membrane lesions within seconds. The annexins form a temporary scaffold at the wound site, holding the edges together while other repair mechanisms complete the job. It's happening constantly, invisibly, in pond water and soil all around us. These tiny beings, continuously repairing themselves, going about their business of living. Before the break, let's return to where we started, that idea that damage is detected before anything truly breaks. Your cells don't wait for catastrophe. They're monitoring stress in real time, feeling the forces being applied to their structures, ready to respond the instant those forces become dangerous. This is fundamentally different from how we often think about healing. We imagine injury as a discrete event, something breaks, then healing begins. But at the cellular level, it's more nuanced. There's a gradient, a spectrum between normal stress and dangerous damage, and cells are navigating this spectrum constantly. The LIM domain proteins stretching with the cytoskeleton, the calcium sensing systems poised to trigger repair, the vesicles floating nearby ready to patch membranes. All of this exists in a state of readiness, a quiet vigilance that never sleeps. Right now, as you listen to these words, cells throughout your body are performing these invisible miracles. Some are repairing minor damage you'll never know occurred. Others are simply maintaining themselves, their sensing systems confirming that all is well, that the forces being applied are within normal ranges. The poetry of repair. There's something almost meditative about contemplating these processes. The precision, the speed, the elegant simplicity of the solutions that evolution has crafted over billions of years. A hole appears in a membrane, a disaster on a cellular scale. Calcium rushes in, and rather than being merely destructive, this influx becomes a signal. Vesicles respond, membranes fuse, and within seconds, integrity is restored. It's happening in the cells of your skin as you shift position, in the cells lining your blood vessels as your heart beats, in the cells of your muscles as you breathe. Constant micro-repairs, constant vigilance, constant restoration. You are not a static thing, frozen in the form you woke up with this morning. You are a dynamic process, a continuous act of self-maintenance and repair, operating at every scale from the molecular to the organism. As we continue our journey tonight, we'll explore how this cellular wisdom scales up, how individual cells communicate damage to their neighbors, how tissues coordinate repair, how the body as a whole responds to injury with systems layered upon systems, each as elegant as these first molecular responses we've explored. But for now, rest in this knowledge. You are being maintained, moment by moment, by processes older than mountains, more reliable than the tides. Your cells know how to heal themselves. They've known for billions of years. And they're doing it right now, quietly, efficiently, while you simply exist. Breathe slowly. Let this settle into your awareness. You are, in every moment, being taken care of from within. The price of perfection. Living with constant repair. Imagine a grand old library, the kind with soaring ceilings, endless rows of books, and that particular smell of aged paper and leather bindings. Now imagine that this library contains not thousands, but billions of volumes. And every single day, some of these books are damaged. Pages torn. Words smudged. Entire sentences corrupted by water damage or fading ink. This library is your genome. And the damage. It's happening right now. The daily assault. We often think of our DNA as something permanent, a stable blueprint locked away safely in the nucleus of every cell. But this image, comforting as it is, misses something fundamental about the nature of biological existence. Your DNA is under constant assault. Each cell in your body, and you have roughly 37 trillion of them, accumulates tens of thousands of DNA lesions every single day. Not over a lifetime. Not over a year. Every day. These lesions come from everywhere. From normal metabolic processes that generate reactive molecules as byproducts. From radiation, cosmic rays from space, natural radioactivity from the Earth, even the gentle sunlight streaming through your window. From heat itself, which causes chemical bonds to occasionally break just from random thermal motion. Your DNA, that molecule that makes you uniquely you, is being damaged constantly, relentlessly, as an inevitable consequence of being alive. And yet, you're fine. You're more than fine. You're here, listening, thinking, being. Your cells are functioning. Your genes are expressing themselves properly. Your body is maintaining its extraordinary complexity. How? The silent guardians. The answer lies in one of biology's most underappreciated miracles: continuous repair. Inside each of your cells, at this very moment, specialized enzymes are patrolling your DNA. They're moving along the double helix, checking, sensing, probing for abnormalities. When they find damage, and they find it constantly, they stop and begin their work. These repair enzymes are exquisitely sensitive. They can detect a single incorrect base pair among the 3 billion base pairs in your genome. They can sense when the DNA structure is slightly distorted, when chemical groups have been added that shouldn't be there, when bonds have broken that should be intact. Think about that sensitivity for a moment. It's like being able to find a single typo in a library containing 1,000 copies of the Encyclopaedia Britannica, where the typo could be as subtle as a missing serif on a single letter. And these enzymes don't just detect problems, they fix them. Some cut out damaged sections and synthesize new DNA to fill the gap. Others reverse chemical modifications, restoring damaged bases to their correct form. Still others coordinate complex processes involving multiple proteins, all working together to maintain the integrity of your genetic information. This repair work never stops. Not while you're awake. Not while you sleep. Not for a single moment from the time a cell is born until the time it dies. Repair is not exceptional. Repair is the norm. Damage is the given, and constant vigilance is the price of staying alive. The Altered Book. But here's something profound, something that changes how we should think about healing and repair. When DNA is repaired, it's never exactly the same afterward. Imagine repairing that torn page in our imaginary library. You carefully glue the pieces back together, press them flat, smooth out the wrinkles. The words are all there. The information is preserved. But the page itself? It's different now. Slightly thicker where the glue dried. A faint crease where the tear was. The paper fiber disrupted, its texture changed. DNA repair is similar, but with consequences that ripple outward in ways both subtle and profound. When a section of DNA is repaired, the three-dimensional structure of the chromatin, the complex of DNA and proteins that packages your genome, is altered. The DNA might fold slightly differently. Proteins might bind with different affinities. Regions that were accessible to the cellular machinery might become less so, or vice versa. These changes affect gene activity. A gene that was being actively read might become quieter. One that was dormant might become more responsive. The cell doesn't just restore its DNA, it emerges from the repair process subtly transformed. This is not a flaw in the repair system. It's an inevitable consequence of the fact that structure and function are inseparable at the molecular scale. You cannot fix something without touching it, and you cannot touch something at this scale without changing it. The cost of staying whole. And there's another cost, perhaps even more fundamental: energy. DNA repair requires energy. A lot of it. Each time an enzyme finds and fixes a DNA lesion, the cell must invest ATP, the universal energy currency of life. It must synthesize new DNA bases to replace damaged ones. It must recruit and coordinate multiple proteins. It must pause other activities to focus resources on the repair. Multiply this by tens of thousands of repair events per day, per cell. Then multiply by 37 trillion cells. The total energy investment is staggering. This energy comes from somewhere. It's diverted from other processes. From growth, perhaps. From responding to signals from other cells. From movement or synthesis, or any of the thousands of other things a cell might be doing. A cell focused on repair is, in some sense, a cell withdrawn from the world. The quieting. Scientists have observed something remarkable in cells that have undergone extensive DNA repair. These successfully repaired cells, cells that have weathered damage and emerged with their genomes intact, can become less responsive to stimuli. They're still alive. Their DNA is functional. But they're quieter somehow, as if the experience of repair has left them more cautious, more inward-focused. It makes a certain kind of sense, doesn't it? A cell that has just invested enormous resources into maintaining its integrity might not be in the best position to respond vigorously to new challenges. It needs time to recover, to rebuild its energy reserves, to return to full function. This is the hidden cost of repair that we rarely consider. Healing takes something from us, even as it preserves us. We emerge intact, but changed. Protected, but diminished in some small way. The meditation of maintenance. There's something deeply philosophical about this constant repair, this endless cycle of damage and restoration. We like to think of ourselves as stable entities moving through time. We imagine we're the same person we were yesterday, that our cells are the same cells, that our bodies are persistent things rather than processes. But the reality is more beautiful and more strange. You are being constantly damaged and constantly repaired. The DNA in your cells is not the same DNA it was this morning. It's been altered, fixed, changed in tiny ways by thousands of repair events. The proteins that make up your cellular machinery are being damaged by oxidation, by heat, by random chemical reactions, and being replaced continuously. You are not a thing. You are a process of maintenance, a continuous act of self-repair that has been going on since the first moment of your existence and will continue until the last. Right now, this very second, repair enzymes are working in your cells. They're finding damage, excising it, synthesizing new DNA, restoring integrity. They're using your energy, changing your chromatin structure, subtly altering which genes are active and which are quiet. And this is normal. This is what being alive means. The cost we don't see. But let's sit with that cost for a moment. Every time your body repairs itself, it pays a price. Energy that could have gone elsewhere. Subtle changes in gene expression that accumulate over time. Cells that emerge from repair slightly less responsive, slightly more withdrawn. This is the bargain of biological existence. You can stay whole, but you cannot stay unchanged. You can be repaired, but you cannot be perfectly restored to what you were before. And yet, somehow, this works. Somehow, despite the costs, despite the imperfections, despite the fact that repair always changes what it fixes, somehow, you persist. Your genome remains functional across decades. Your cells coordinate into tissues. Your tissues form organs. Your organs sustain consciousness, thought, feeling, life. The repair is good enough. Not perfect, but good enough. And good enough, in biology, is often spectacular. The rhythm of restoration. Think of it like maintaining an old house. You fix the roof when it leaks, patch cracks in the walls, replace worn floorboards. Each repair preserves the house, but also changes it slightly. New wood doesn't quite match old. Paint colors shift across years. The house becomes a palimpsest, a layered history of maintenance and care. Your body is the same, but operating at a scale that makes a house seem simple by comparison. Some of your cells are making do with slightly altered chromatin structures from yesterday's repairs. Others are recovering from the energy expenditure of fixing last night's accumulated damage. Still others have entered temporary quiet states, pulling back from full activity while they address their maintenance needs. And somehow, the whole system continues to function. The orchestra plays on, even as individual musicians occasionally miss notes while adjusting their instrument. Dormancy as wisdom. That strategy the yeast employ, entering dormancy when damage becomes overwhelming. Perhaps there's a lesson there for us. Sometimes the wisest response to damage isn't to push through, to maintain normal activity while trying to repair in the background. Sometimes the wisest response is to stop, to withdraw, to dedicate resources fully to healing rather than splitting attention between healing and performance. We see echoes of this in our own biology. The fatigue we feel when fighting an infection. The deep sleep that follows intense physical exertion. The way trauma can leave us withdrawn, turned inward, less responsive to the world while something deeper heals. These aren't failures. They're the same ancient wisdom that allows yeast to survive impossible damage. The recognition that repair requires resources, and sometimes the most survival-oriented choice is to prioritize repair over everything. LSE. The permanent impermanence. As we move deeper into our exploration of repair tonight, carry this understanding with you. You are never the same twice. Every act of repair changes you. Every fixed DNA lesion alters your chromatin landscape. Every cell that withdraws into a quieter state shifts the balance of your body's function. You are being maintained, yes, but maintenance and transformation are the same process. This isn't something to fear. It's the fundamental nature of living systems. We are patterns that persist not by staying the same, but by continuously changing in response to damage while somehow preserving our essential organization. You are a river, not a stone. Always flowing, always changing, yet recognizably you. Ralph. The enzymes continue their patrol. The repair proceeds. The costs accumulate, and the benefits accumulate faster. Life continues its ancient bargain. Stay whole enough, adapt constantly, and persist through time not by resisting change but by managing it. Your DNA is being damaged right now. And right now, it's being repaired. This is not a crisis. This is Tuesday. This is every moment of every day. This is what it means to be alive. Breathe slowly. Feel the quiet work happening within you. Trillions of repair events, each one slightly changing you, each one keeping you whole. You are being maintained. You are being transformed. These are the same thing. And it's enough. It's always been enough. The art of self-renewal. Single cells as master builders. Picture an artist who creates a sculpture, then deliberately breaks it, then rebuilds it, all within the span of a lunch break. Now imagine that artist is microscopic, has no brain, no hands, no conscious intent. And yet the sculpture they create is more intricate than anything in the Louvre, and they rebuild it perfectly, dozens of times a day, for their entire existence. Welcome to the world of single-celled organisms that rebuild themselves. The 2-hour miracle. In ponds and puddles, in drops of water clinging to leaves, in the soil beneath your feet, single cells are performing acts of regeneration that would seem miraculous if we could witness them at our scale. Some of these organisms can regrow entire lost structures, complex, functional parts of their cellular bodies, in under 2 hours. 2 hours. The time it takes to watch a movie. The time between lunch and mid-afternoon. And in that brief span, a cell no larger than a grain of pollen can reconstruct machinery so sophisticated that humanity, with all our technology, cannot replicate it. Let's start with a particular favorite of biologists, Chlamydomonas, a single-celled green alga that moves through water using whip-like appendages called flagella. These flagella are marvels of biological engineering. Each one contains hundreds of proteins arranged in precise geometric patterns. They beat in coordinated waves, propelling the cell through its liquid world at speeds that would translate, at human scale, to a brisk run. And when conditions become stressful, when the environment becomes toxic, when the cell needs to conserve energy, Chlamydomonas simply sheds its flagella. Deliberately. It lets them break off and drift away. This seems almost reckless, doesn't it? Like throwing away your legs because you're tired. But here's the astonishing part. Within approximately 90 minutes, the cell regrows them completely. The choreography of reconstruction. Imagine what this requires. The cell must synthesize hundreds of different proteins in precisely the right amounts. It must transport these proteins to the assembly site at the cell's surface. It must arrange them in the correct three-dimensional configuration, building from the base upward, extending the flagellum outward like a tower being constructed from the ground up. All of this happens without blueprints in the way we understand them, without a foreman directing traffic, without a project manager coordinating the work. The cell simply does it. The information for how to rebuild is distributed throughout the system, in the proteins themselves, in the way they naturally interact, in the cellular machinery that coordinates their synthesis and transport. And 90 minutes later, the cell is whole again. It can move. It can seek light for photosynthesis. It can navigate its microscopic world. The cell that shed its flagella in response to stress is not the same cell that grows them back. It's been through a cycle of dismantling and reconstruction. And yet it remains itself, its essential identity preserved through the transformation. Breaking toward the light. But some cells take this rebuilding strategy to an even more extreme level. Consider what happens during photosynthesis, the fundamental process by which plants, algae, and some bacteria convert sunlight into chemical energy. At the heart of this process sits a protein complex called photosystem II. This molecular machine does something almost absurdly difficult. It splits water molecules, tearing apart their bonds to extract electrons. These electrons flow through a series of steps that eventually store energy in chemical form. But splitting water creates byproducts, highly reactive oxygen species that damage proteins. And photosystem II, because it's doing the water splitting, takes the brunt of this damage. It's like standing at the forge, closest to the flames, taking the full heat. Under strong light, the very condition where photosynthesis is most productive, photosystem II is continuously breaking down. Not occasionally. Continuously. So what does the cell do? It continuously rebuilds. The perpetual reconstruction. Think about this for a moment. The protein complex that captures light energy and powers the most important biochemical process on Earth cannot survive the very process it enables. It's like having an engine that destroys itself in the act of running. And yet, it works. It works because cells have evolved a solution more elegant than building an indestructible photosystem. They've mastered the art of perpetual reconstruction. Throughout the day, as sunlight pours down, as photosynthesis proceeds, photosystem II complexes are being disassembled and rebuilt. Damaged components are removed. New ones are synthesized and inserted. The machine is repaired while it's running, like changing the tires on a moving car. This is happening right now in every green leaf, every blade of grass, every floating algal cell. The machinery of photosynthesis is breaking and being rebuilt, breaking and being rebuilt, in an endless cycle. The leaf doesn't stop being a leaf. The grass doesn't stop being grass. But at the molecular level, they are in constant flux, continuously self-renewing, maintaining function through perpetual reconstruction rather than through durability. Lighty. Glass houses that heal. And then there are the diatoms. Diatoms are single-celled algae that live inside intricate glass houses, shells made of silica, the same material as sand and quartz. These shells are masterpieces of natural architecture, covered in elaborate patterns of holes and ridges, each species creating its own distinctive design. Building these shells requires the cell to manipulate silicon chemistry in ways that seem almost magical. They precipitate silica from solution, shaping it into precise three-dimensional structures, creating forms of incredible beauty and complexity. And when these glass houses are punctured, when damage occurs to the shell that protects and defines them, diatoms can reseal the breach. They're repairing glass. At room temperature. In water. With no furnace, no glass blowing, just biological processes we're still trying to fully understand. The diatom detects the damage, mobilizes silica-containing compounds to the wound site, and precipitates new glass to fill the gap. The shell is made whole again, its protective function restored. A single cell, repairing its glass house, beneath the microscope, in the silent depths of the ocean. Sacrifice and protection. Some single cells take their repair responses a step further, they protect their neighbors. Cyanobacteria, the ancient photosynthetic bacteria that first filled Earth's atmosphere with oxygen billions of years ago, sometimes face conditions where certain nutrients become scarce. Specifically, when nitrogen is limited, These bacteria face a problem. They need nitrogen to build proteins, but the nitrogen needed for protein synthesis competes with the nitrogen needed for photosynthesis. Some cyanobacteria solve this through differentiation. Certain cells in a chain of cyanobacteria transform into specialized cells called heterocysts. These cells stop photosynthesizing and instead focus on nitrogen fixation, converting atmospheric nitrogen into a form the other cells can use. But here's what's remarkable. These heterocyst cells thicken their walls, create protective layers, and essentially sacrifice their own growth and reproduction to perform a service for their neighbors. They're responding to stress not by withdrawing into themselves, but by transforming into something new, something that protects and provides for the community of cells they're part of. A single cell, sensing stress, choosing to become a guardian. Explosive healing. The single-celled organism Paramecium, that slipper-shaped microbe you might remember from biology class, has evolved one of the most dramatic wound sealing mechanisms in the microscopic world. Embedded throughout a paramecium's outer layer are thousands of tiny dart-like structures called trichocysts. Under normal conditions, they sit quietly, loaded with calcium like tiny compressed springs. But when the cell is wounded, when its membrane is breached, calcium from outside rushes in, just as we discussed earlier. And this calcium influx triggers the trichocysts near the wound to explosively discharge. They fire outward in a coordinated burst, releasing their contents and creating, in that instant, a patch that helps seal the wound. It's violent, immediate, effective, an explosive healing response that happens faster than you can blink. The paramecium doesn't think about this. It doesn't plan it. The damage itself triggers the response, and the response is so fast, so coordinated, that the cell often survives injuries that would seem catastrophic. The coat of many colors. Then there are parasites like trypanosomes, the single-celled organisms that cause sleeping sickness. These cells face a particular challenge. They live inside mammalian hosts whose immune systems are constantly trying to destroy them. The host immune system recognizes the proteins on the trypanosome's surface and mounts an attack. You'd think this would be the end of the parasite. But trypanosomes have evolved a remarkable strategy. They regularly shed their entire surface coat and rebuild it with completely different proteins. It's like a fugitive constantly changing disguises. By the time the immune system has recognized one surface coat and mobilized to attack it, the trypanosome has already shed that coat and grown a new one. The antibodies the immune system created are suddenly useless, designed to recognize a surface that no longer exists. This continuous rebuilding allows the parasite to survive in an actively hostile environment. It's dodging destruction by constantly becoming, at least superficially, a different cell. The trypanosome that enters your bloodstream is not the same trypanosome that's still there weeks later, not in terms of its surface, anyway. It's been rebuilt, transformed, renewed, again and again. From a single spark. Perhaps the most astonishing example of single-cell regeneration comes from a beautiful organism called Acetabularia, a single-celled alga that looks like a tiny umbrella, growing several centimeters tall in shallow Mediterranean waters. Despite its complex shape, a stem-like stalk topped with a delicate cap, Acetabularia is a single cell. One membrane. One cell. But with a nucleus positioned at the base of the stalk. And if you cut away everything except that basal nucleus, the cell will regenerate its entire body. The entire visible structure, stalk, cap, all of it, will grow back from just the nucleus and a small bit of surrounding cytoplasm. It's difficult to overstate how remarkable this is. The nucleus, that small repository of genetic information, contains somehow enough information to rebuild the entire complex architecture of the cell. And the cell, responding to nothing but its own internal programming, does exactly that. From a fragment, wholeness to RNS. Defensive architecture. Even bacteria, The simplest free-living cells have evolved sophisticated self-repair strategies. When bacteria are under attack, when their cell walls are being damaged by antimicrobial compounds or environmental stress, they don't passively accept destruction. Some bacteria can flip lipid components in their cell walls, reorienting defensive molecules to the outer surface, creating a barrier against whatever is attacking them. It's like rotating armor plating to present the strongest surface to incoming fire. This isn't passive chemistry. It's an active response, requiring energy and coordination. The bacterium senses the attack and restructures its own outer boundary to resist it. A cell that has no nervous system, no brain, no consciousness as we understand it, nonetheless responds to threats by remodeling its own arc height. The pattern that persists. What all these examples share is a fundamental principle. Single cells maintain their identity not through rigidity, but through the ability to rebuild themselves. They shed flagella and regrow them. They continuously disassemble and reassemble their photosystems. They repair glass shells and explosive seal wounds. They change their surface coats and regenerate entire bodies from nuclei. They are not static entities. They are processes, patterns of organization that persist through constant material turnover. The Chlamydomonas with newly regrown flagella is, in some sense, not the same cell that shed them 90 minutes ago. Materials have been exchanged, structures have been rebuilt. And yet it is the same cell. The pattern persists. The organization maintains itself. This is true for all living things, but it's most visible in these single cells, where the entire drama of dismantling and reconstruction plays out in hours or minutes rather than years. Renewal as survival. There's something almost meditative about these single cells continuously rebuilding themselves. They're not trying to achieve immortality through permanence. They're achieving persistence through renewal. They survive not by resisting change, but by embracing it, by tearing down and building up, by shedding and regrowing, by breaking and repairing. Right now, in the water systems around the world, in soil, in the ocean, trillions of these cells are engaged in their cycles of reconstruction. Chlamydomonas are regrowing flagella. Photosystems are being rebuilt under the light. Diatoms are sealing their glass shells. Cyanobacteria are forming protective cells. Paramecium are explosively patching wounds. Trypanosomes are changing their coats. Acetabularia are regenerating entire bodies. None of them know they're doing this. It simply happens, emerging from the logic of their molecular organization, from billions of years of evolutionary refinement. They are, each one, a tiny demonstration of life's fundamental strategy. Maintain the pattern, not the parts. Preserve the organization, not the materials. Be willing to tear down and rebuild, because flexibility is more valuable than rigidity. As we move forward in our exploration tonight, we'll see how these same principles scale up, how multicellular organisms coordinate the repair and renewal of millions or billions of cells, how tissues regenerate, how whole organisms heal. But the foundation is here, in these single cells that rebuild themselves in hours or minutes. They're teaching us something profound. You don't have to stay the same to remain yourself. The cell that sheds its flagella and regrows them is still the same cell. The photosystem that's rebuilt every few hours still captures light. The acetabularia that regenerates from its nucleus alone is still acetabularia. Identity persists through transformation. Organization maintains itself through flux. You are like these cells, scaled up and slowed down. You are continuously breaking down and rebuilding, shedding and regrowing, repairing and renewing. The you of this morning is not, at the molecular level, quite the same you listening now. Proteins have been replaced. Membranes have been repaired. Structures have been rebuilt. And yet you remain yourself. The pattern persists. Breathe slowly. Feel yourself as a process rather than a thing. A pattern of organization maintaining itself through continuous renewal. You are always becoming yourself. You are never finished. And that's exactly as it should be. The architects of impossibility, bodies that rebuild themselves. There's a story, perhaps apocryphal, about a Japanese art form called kintsugi, where broken pottery is repaired with lacquer mixed with gold. The breaks aren't hidden, they're highlighted, celebrated. The repaired vessel is considered more beautiful for having been broken. But imagine if the bowl could repair itself. Not just seal the cracks, but regrow the missing pieces. Imagine if you could shatter it into a dozen fragments, and each fragment could rebuild an entire bowl. This isn't poetry. This isn't metaphor. For some organisms, this is simply Tuesday. Beyond repair. We've been exploring repair, the patching of wounds, the fixing of damage, the restoration of what was. But now we're going to venture into territory that challenges our very understanding of what a body is. We're entering the realm of regeneration. Not repair, but regeneration. Not patching, but rebuilding. Not restoring, but recreating. Some organisms on this planet can regrow entire bodies from fragments. Not small wounds. Not damaged tissues. Entire bodies. Complete organ systems. Hearts, brains, digestive tracts, sensory structures, all rebuilt from a piece of tissue smaller than your fingernail. This seems impossible. It violates our intuitions about what bodies are, how they work, how they maintain their form and function. And yet it happens, constantly, in oceans and streams and tide pools around the world. The sea squirt's secret. Let's begin with creatures you've probably never heard of. Sea squirts, or tunicates, strange, vase-like animals that attach to rocks and filter feed from the ocean current. They don't look like much. Adult sea squirts are sessile, stationary, looking more like rubbery sacks than animals. If you didn't know better, you might mistake them for sponges or corals or some kind of plant. But sea squirts are animals. They're chordates, actually, they belong to the same broad group as fish, birds, and mammals. In their larval stage, they even have a notochord, the precursor to a backbone. And they possess an ability that would seem like magic if we encountered it in a creature we understood better. Cut a sea squirt into small pieces containing just a bit of various tissues, and those pieces can regenerate all organs. Not heal the wound. Not repair the damage. Regenerate all organs. Building a heart from memory. Consider Polycarpa mytoligera, a sea squirt that lives in shallow Mediterranean waters. Researchers have taken small fragments of this animal, pieces that contained no heart, no neural structures, no complete organs of any kind, and watched what happened. The fragment doesn't die. It doesn't simply seal its wounds and persist as a partial creature. It rebuilds. Over days and weeks, the fragment develops a new heart. The cardiac tissue organizes itself, begins beating, establishes circulation. Neural structures reform, rebuilding the simple nerve networks the animal needs to sense and respond to its environment. The fragment knows, somehow, what the whole animal should look like. And it builds toward that template. Think about what this means. The information for the entire body plan, for where the heart should be, how the nervous system should be organized, which tissues need to connect to which others, all of this information is present in a small fragment. It's not centralized in a brain, because sea squirts barely have brains. It's not stored in a single location. It's distributed somehow throughout the tissues, encoded in patterns of gene expression, in the way cells signal to each other, in gradients of molecules that tell cells where they are and what they should become. The fragment is not trying to survive as a fragment. It's trying to become whole again. The democracy of stem cells. To understand how this is possible, let's turn to one of science's favorite regeneration champions, the planarian flatworm. These tiny creatures, found in freshwater streams and ponds, have fascinated biologists for centuries. Cut one in half, and each half regenerates into a complete worm. Cut it into pieces, and each piece attempts to regenerate the whole animal. The secret to their regenerative power lies in a remarkable commitment. Planarians devote approximately 15% of their entire body mass to stem cells. 15%. Imagine if 15% of your body consisted of cells that could become anything: neurons, muscle, digestive tissue, whatever was needed. These stem cells, called neoblasts, are distributed throughout the planarian's body. They're waiting, in some sense, for their moment. And when damage occurs, when the worm is cut, they spring into action. They migrate to the wound site. They begin dividing. They start differentiating into specialized cell types, building whatever structures are missing. The miracle of bilateral regeneration. Here's where it gets truly extraordinary. If you cut a planarian in half, head on one side, tail on the other, both pieces survive and regenerate. The head piece regenerates a new tail. That makes intuitive sense, perhaps. The brain and sensory organs are preserved, and the body grows back. But the tail piece regenerates a new head, a new brain, new eyes, new sensory organs. The cells in the tail somehow know they need to build a head. They organize themselves into the complex three-dimensional structure of a planarian brain. They form photoreceptor cells that will become eyes. They establish the neural networks necessary for the worm to sense and move through its environment. Two worms from one. Both complete. Both functional. Both capable of feeding, moving, reproducing, and, if cut again, regenerating once more. The cut worm doesn't die. It multiplies. The question of identity. This raises a philosophical puzzle that's worth sitting with for a moment. Which of these two regenerated worms is the original? The head that grew a new tail? The tail that grew a new head? Both. Neither. The planarian that was cut no longer exists, in a sense. Two new planarians exist instead, each carrying forward aspects of the original but also fundamentally transformed by the regeneration process. The headpiece worm has new body tissues, new tail structures, cells that didn't exist before the cut. The tailpiece worm has a new brain, new memories forming, new sensory experiences, a new neural organization. Are they the same worm? Are they siblings? Are they something we don't have good words for? The planarian doesn't care about these questions. It simply regenerates, becoming whole through a process so reliable that it's been doing it for millions of years. The ultimate regeneration. But planarians, remarkable as they are, aren't even the most extreme regenerators. That honor might belong to sponges. Sponges are animals, though they barely look it. They have no nervous system, no muscles, no organs in the conventional sense. They're assemblages of cells working together, filtering water to extract nutrients. And if you take a sponge and push it through a fine mesh screen, literally dissociating it into individual cells, destroying every connection, every structure, every organizational feature, those cells will find each other again. They reaggregate. They reform connections. They rebuild the sponge. It's as if you took apart a car, separated every single component down to individual nuts and bolts, scattered them across a parking lot, and they spontaneously reassembled themselves into a working vehicle. Except it's more miraculous than that, because the sponge cells don't have instructions printed on them. They don't have a blueprint to follow. They have only their own ability to recognize other sponge cells, to adhere to them in appropriate ways, to establish the patterns of organization that make a functional sponge. The information for the whole exists somehow in the properties of the parts and the ways they interact. Reorganizing reality. Hydra, those tiny freshwater polyps with tentacles that wave in the current, take a different approach to regeneration. Cut a hydra in half, and you don't get two separate hydra, though that can happen too. What you often get is a single hydra that reorganizes its entire body plan. The two halves fuse back together, but in a new configuration. The head might be in a different position. The foot, the structure that attaches the hydra to surfaces, might relocate. The entire body plan shifts, adapts, reorganizes. It's as if the hydra doesn't have a fixed body plan so much as a set of organizational principles that can be applied flexibly depending on what tissue is available. This hydra exists at a given moment in a particular configuration. But that configuration is contingent, flexible, revisable. If circumstances change, if the body is bisected, the organizational principles assert themselves again, and a body plan emerges from the available materials. The hydra is less like a building and more like a dance, a pattern that persists through constant reorganization of its components. The reset button. Some jellyfish have evolved perhaps the most radical regeneration strategy of all. They can reverse their development and start over. Certain species, when stressed or damaged, can transform their adult bodies back into juvenile forms, essentially rebooting their entire life cycle. It's called transdifferentiation. Cells that had specialized into one type transform into another type, or even back into stem cell-like states. The adult jellyfish essentially dissolves its own complexity and rebuilds as a younger version of itself. Imagine if you could respond to injury by becoming a child again. Not metaphorically, literally. Your adult tissues reorganizing, de-differentiating, then following the developmental program again to rebuild an adult body. This is what some jellyfish do. And theoretically, they can do it indefinitely. They're not immortal, they can be eaten, can starve, can die from disease. But aging and damage, those can trigger a reset, a return to youth, another chance at life. The dream of seamless healing, bodies that erase injury. There's a kind of beauty we rarely see in our own bodies, the beauty of healing that leaves no trace. When you cut your finger, a scar forms. When bone breaks, the fracture line remains visible in X-rays years later. When tissue tears, it repairs with fibrous material that's stronger than the original but structurally different, functionally limited. We accept this as the price of healing. We're grateful for scars. They mean we survived, that damage didn't destroy us. We carry them as evidence of resilience. But what if healing could be perfect? What if damage could be erased rather than merely sealed? What if your body could rebuild exactly what was lost, with no trace that injury ever occurred? For most of us, this is fantasy. But for some creatures on Earth, this is simply how healing works. The axolotl's gift. In the lakes and canals around Mexico City, there lives a creature that seems to have stepped out of myth, the axolotl. These salamanders, with their feathery external gills, their perpetual smile, their otherworldly appearance, remain in larval form throughout their lives, never quite growing up in the way most amphibians do. But their arrested development comes with an extraordinary gift. They can regenerate complex structures with no scarring whatsoever. Lose a limb, and the axolotl will regrow it. Not approximate it. Not replace it with scar tissue. Regrow it, complete with bones, muscles, blood vessels, nerves, skin. Every tissue in perfect arrangement. Every structure fully functional. And when regeneration is complete, you cannot tell which limb was lost. There's no scar. No deformity. No evidence that damage ever occurred. The new limb is indistinguishable from the origi. Null. The cellular orchestra. What makes this possible is a process of breathtaking complexity and precision. When an axolotl loses a limb, the cells at the wound site do something remarkable. They de-differentiate. Specialized cells, muscle cells, bone cells, cartilage cells, essentially forget what they are and return to a more stem cell-like state. These dedifferentiated cells form a structure called a blastema, a mass of unspecialized cells that will become the new limb. And then, following cues we're still trying to fully understand, these cells begin to specialize again. But not randomly. They organize themselves into the precise three-dimensional architecture of a limb. Bone cells migrate to where bones should be and begin forming skeletal structures. Muscle cells organize into the proper muscle groups. Blood vessels branch in the correct patterns. Nerves grow along appropriate pathways, reconnecting with the existing nervous system. Skin cells cover the growing structure, forming skin that matches the surrounding tissue perfectly. The blastema knows, somehow, what a complete limb looks like. And it builds toward that template with astonishing fid. Aleti. Repeated miracles. Perhaps even more remarkable, axolotls can do this repeatedly. Lose a limb once, regrow it. Lose the same limb again, regrow it again. There's no limit, at least none that scientists have found. An axolotl can regenerate the same limb dozens of times over its lifetime, each time with the same perfection. This isn't just healing. It's regeneration as a renewable resource, an ability that doesn't diminish with use. The axolotl doesn't run out of regenerative capacity. It doesn't heal more poorly the second or third time. Each regeneration is as perfect as the first. And it's not just limbs. Axolotls can regenerate portions of their heart, parts of their brain, sections of their spinal cord, their eyes, their jaws. Almost any structure can be rebuilt if damaged. They live their lives with a kind of biological insurance policy that seems almost unfair. No matter what happens to their bodies, they can rebuild. The stars of the sea. In tidal pools and on rocky ocean floors, starfish demonstrate another form of scarless regeneration, one even more dramatic in some ways than what axolotls achieve. Many species of starfish can regrow entire arms from small fragments of the central disc. Not just regrow lost arms, that would be impressive enough, but in some species, a single severed arm with a piece of the central disc can regenerate an entire new starfish. The process takes months, but it's astonishing to witness. The fragment first seals its wounds, then begins generating the missing body. Arms sprout. Organs form. The digestive system reorganizes. The water vascular system, the unique hydraulic system that starfish use for movement, rebuilds itself. Eventually, a complete starfish emerges. 5-armed, or 7, or 12, depending on species, fully functional, capable of moving, feeding, reproducing. And when regeneration is complete, there's no scar tissue marking where the old body ended and the new began. The regenerated arms are identical to the originals. The new starfish is a complete starfish, indistinguishable from one that was naive. Are damaged. Hearts that heal themselves. Zebrafish, those tiny striped fish beloved of aquarium enthusiasts and researchers alike, possess an ability that would revolutionize human medicine if we could replicate it. They can regrow heart tissue. Remove up to 20% of a zebrafish heart, and it will regenerate the missing portion. The heart continues beating throughout the process, maintaining circulation even as it rebuilds itself. The secret lies in the zebrafish's ability to reactivate embryonic genes, genetic programs that normally operate only during development, when organs are forming for the first time. When heart damage occurs, surviving heart muscle cells essentially roll back their developmental clock. They begin dividing, something adult mammalian heart cells almost never do. They proliferate, forming new cardiac tissue that integrates seamlessly with the existing heart. The regenerated tissue doesn't just fill the gap with scar, it forms functional heart muscle that contracts in coordination with the rest of the organ. Blood vessels grow into it. Electrical signals propagate through it properly. Within weeks, the heart is whole again. And unlike a human heart recovering from a heart attack, where scar tissue permanently reduces function, the zebrafish heart returns to Fu. LL capacity. The tadpole's tail. African clawed frogs present a fascinating case study in the limits and possibilities of regeneration. As tadpoles, these frogs can regenerate their tails perfectly. Sever the tail, and it regrows, complete with spinal cord, muscle, skin, blood vessels. The regenerated tail is fully functional and scarless. The key to this regeneration is a signaling pathway called WNT signaling, a cascade of molecular communications that tells cells at the wound site to begin building a new tail. But here's what's intriguing: this ability is temporary. As the tadpole metamorphoses into an adult frog, it loses most of its regenerative capacity. Adult African clawed frogs can heal wounds, but they can't regenerate lost limbs or organs without scarring. Something about the developmental transition from tadpole to frog switches off the regenerative programs. The genetic potential remains, the genes are still there, but they're no longer activated in response to injury. This suggests something profound. The difference between scarring and scarless regeneration isn't necessarily about having different genes. It's about which genes are turned on or off, which developmental programs are accessible in response to injury. The tadpole and the adult frog have essentially the same genome, but they heal differently. The potential for perfect regeneration exists in the adult, it's just suppressed somehow. Arms of the octopus. Octopuses, those alien intelligences sharing our planet, can regrow portions of their arms if the tips are damaged or lost. And these aren't simple structures being regenerated. Octopus arms contain complex nervous systems. A significant portion of an octopus's neurons are actually in its arms rather than its brain. The arms also contain intricate muscles, blood vessels, and hundreds of suckers, each one a sophisticated sensory and motor organ. Yet when arm tips are damaged, octopuses can regenerate them. The nerves reconnect properly. The suckers reform with their complex internal structures intact. The regenerated tissue integrates seamlessly with the existing arm. Once healed, the regenerated section functions normally. The octopus can control it with the same dexterity as before. The suckers grip and taste and sense as they should. The arm tip is, for all practical purposes, perfect. This is regeneration of neural tissue, something mammals do very poorly, accomplished by an invertebrate with what seems like casual ease. The escape artists. In the deserts and arid regions of Africa, there lives a mouse with a strategy so bizarre it seems like evolutionary desperation. When caught by a predator, African spiny mice can shed their skin. Not just a little skin. Large patches, up to 60% of the skin on their backs, can tear away cleanly, leaving the predator with a mouthful of skin while the mouse escapes. This seems like a catastrophic injury. Exposed muscle, no protective barrier, massive risk of infection and water loss. But these mice regrow the lost skin. Completely. With hair follicles, sweat glands, even the dermis layer that gives skin its structural strength. And they do it with minimal scarring. The regenerated skin is functional, protective, essentially identical to what was lost. Within weeks, you can barely tell the mouse was ever injured. It's an extreme example of trading acute injury for survival, banking on regenerative capacity to recover what was sacrificed in the escape. Ape. The lizard's bargain. Lizards are famous for dropping their tails when threatened, a process called autotomy. The tail breaks at a predetermined fracture plane, continues twitching to distract the predator, and the lizard escapes. What's less famous is that lizards regrow these tails. Not identical replacements, the regenerated tail typically has a cartilaginous rod instead of proper vertebrae, and the pattern of scales is often different. But it's functional. It helps with balance. It can store fat. It allows the lizard to continue living normally. And the regeneration happens without excessive scarring at the attachment point. The new tail integrates reasonably smoothly with the body, even if it's not a perfect copy of the original. Different species have different degrees of fidelity in their regeneration. Some produce tails that are remarkably similar to the originals. Others produce more obviously regenerated structures. But all of them demonstrate that complex appendages can be rebuilt, that loss doesn't have to be permanent, that even vertebrates, our own group, retain some capacity for true regeneration. Bodies in flux. Sea anemones, those flower-like animals anchored to rocks in tide pools, can remodel their entire bodies after injury. Tear one in half, and instead of 2 dying fragments, you often get 2 anemones. Each piece reorganizes, reforms its oral disc, the tentacle-bearing end, re-establishes its foot, the attachment structure, and continues living. The process is fluid, organic, almost sculptural. The anemone doesn't follow a rigid plan. It adapts to what tissue is available and organizes it into the most functional configuration possible. Tentacles might be fewer or more numerous after regeneration. The body might be smaller initially. But the essential organization, the basic body plan of an anemone, reasserts itself. And there's no scarring. The regenerated tissue is simply anemone tissue, indistinguishable from the original once regeneration is complete. The moss's secret. Even plants demonstrate scarless regeneration, though we sometimes overlook it because we think of plant growth differently than animal healing. Mosses can regenerate entire plants from single leaf cells. Take a single cell from a moss leaf, culture it properly, and it can develop into a whole new moss plant, complete with leaves, stems, the works. This is totipotency taken to an extreme. The idea that every cell contains the complete information needed to build the whole organism, and under the right conditions, can do so. The regenerated moss isn't approximate. It's not a simplified version or a scarred version. It's just moss, as complete and functional as the plant it came from. For mosses, the boundaries between repair, regeneration, and reproduction blur into each other. Growing new tissue, replacing damaged tissue, and creating new individuals are all variations on the same basic process. The rarity of perfection. But here's what's crucial to understand. Scarless regeneration is rare. For every axolotl that regrows a perfect limb, there are thousands of species that heal with scars. For every zebrafish that regenerates heart tissue, there are countless species whose hearts scar after injury. Humans are among the majority. We heal, but we don't regenerate. We seal wounds, but we scar. We survive damage, but we carry its marks. Perfect repair is possible. We know this because some organisms achieve it. But it's not the norm. It's not even common among vertebrates. The trade-off. One possibility is that scarless regeneration requires trade-offs that most complex organisms can't afford. Maintaining the cellular plasticity needed for perfect regeneration might be incompatible with high metabolic rates, or with complex immune systems, or with the sophisticated cellular specializations that enable things like warm-bloodedness, complex cognition, or rapid growth. Axolotls remain in a larval state their entire lives. Zebrafish are small, simple. Starfish are invertebrates with very different body organizations than ours. The organisms that regenerate perfectly tend to be either relatively simple, or permanently juvenile, or cold-blooded, or some combination of these. Perhaps perfect regeneration is a capability that evolution trades away when optimizing for other things. Things like size, speed, intelligence, metabolic capacity, or parental investment in fewer offspring. The suppressed potential. The case of the African clawed frog is particularly tantalizing. The tadpole can regenerate. The adult cannot. Same organism, same genes, different abilities. This suggests that mammals, including humans, might have latent regenerative abilities that are suppressed. The genetic toolkit might be there, just not activated properly in response to adult injury. Some researchers are exploring whether we could reactivate these pathways. Whether we could, essentially, trick adult mammalian cells into behaving like axolotl cells or tadpole cells when injury occurs. It's early work. There are enormous challenges. But the existence of scarless regeneration in some vertebrates proves it's biologically possible. The axolotl isn't doing anything that violates the laws of physics or chemistry. It's just using the tools of cellular and molecular biology in a way that produces perfect regeneration. If one vertebrate can do it, perhaps others could too, given the right conditio. NS. The comfort of scars. But there's something to be said for scars, too. They form quickly. They seal wounds in days rather than weeks or months. They're metabolically cheaper than perfect regeneration. They allow organisms to survive damage and continue living without the extended vulnerability that comes with slow, perfect rebuilding. Scar tissue is evolution's pragmatic solution, not perfect, but fast and reliable. For a mammal living a fast-paced life, needing to escape predators or find food or care for young, the speed of scarring might outweigh the perfection of regeneration. Better to heal imperfectly and survive than to regenerate perfectly while vulnerable. Somewhere in a lake, an axolotl is regrowing a perfect limb. On the ocean floor, a starfish is regenerating an arm. In a laboratory tank, a zebrafish heart is rebuilding itself. In a terrarium, a tadpole tail is reforming without a trace of damage. They're demonstrating what's possible. They're showing us that perfect repair isn't fantasy. It's real, achievable, written into the biological potential of life on Earth. Most of us will never regenerate a limb or regrow heart tissue. We'll heal with scars, repair with fibrosis, live with the marks of our injuries. But knowing that some creatures erase their wounds completely changes how we think about healing. It transforms scarring from an immutable law of biology into a particular strategy, one choice among several that evolution has explored. You carry scars. Physical ones, perhaps. Certainly molecular ones, accumulated cellular damage, repaired DNA, tissues that have healed imperfectly. These scars are not failures. They're evidence of survival. They're the price your body paid to keep you alive in a universe that's constantly trying to disorder you. But the existence of scarless regeneration whispers a possibility. Perhaps this isn't the only way. Perhaps, someday, we'll understand regeneration well enough to coax our own bodies toward perfection. Until then, breathe slowly. Feel your scars, not as defects but as victories. Each one is a place where your body chose survival over perfection, speed over flawlessness. Each one is proof that you're still here. And that, in the end, might be more important than perfection. The Quiet Revolution. How human bodies maintain themselves. The Vidi. Oh. Right now, as you listen to these words, you are shedding. Millions of skin cells are detaching from your body, invisible flakes of dead tissue falling away like autumn leaves. By the end of this month, you will have shed roughly 1 billion cells from your skin alone. This isn't decay. This isn't damage. This is renewal. Your body is running a never-ending program of self-repair and replacement, so constant and so thorough that you are, quite literally, not made of the same physical materials you were made of a few years ago. You cannot regenerate like an axolotl. You cannot regrow a lost limb like a starfish. But you are, in your own way, continuously regenerating yourself at a scale that's both more subtle and more comprehensive than you might realize. The skin you're in. Let's start at your surface, the boundary between you and the world, your skin. Your skin is not a static barrier. It's a living frontier, constantly dying and being reborn. At the deepest layer of your epidermis, cells are dividing. New skin cells are being born every moment, pushing upward through the layers above them. As they rise, they flatten, fill with keratin, lose their organelles, and eventually die, transforming from living cells into protective dead material. This journey from birth to death takes about a month. By the time a skin cell reaches the surface, it's already dead, part of a protective layer of deceased cells that shields the living tissue beneath. And then it falls away. Sloughed off by friction, by washing, by the simple mechanics of living in a body that moves through the world. You don't notice this. It happens too slowly, at too small a scale. But it's happening constantly, everywhere on your body. By some estimates, you shed between 30,000 and 40,000 dead skin cells every hour. Every hour. The dust in your home is largely dead skin cells, yours and those of anyone else who lives there. You are, quite literally, gradually dispersing yourself into your environment, even as you regenerate what you've lost. The cascade of healing. Now imagine you cut your finger. It's a small wound, a cooking accident, nothing serious. The moment your skin is breached, your body initiates a response so coordinated, so complex, that it rivals any military operation in its precision and timing. First, within seconds, blood vessels constrict to minimize blood loss. Platelets, tiny cell fragments circulating in your blood, rush to the wound site. They begin sticking to the damaged tissue and to each other, forming a temporary plug. These platelets release chemical signals that activate a cascade of clotting factors, a sequence of proteins that amplify the signal, each one activating the next, building toward the formation of fibrin. Fibrin forms a mesh that traps blood cells, creating a clot that seals the wound. This all happens in minutes. The bleeding stops. The barrier is sealed, temporarily. But the real work is just beginning. The reconstruction. Beneath the forming scab, your skin is rebuilding. Immune cells migrate to the wound, clearing away debris and dead tissue, fighting any bacteria that might have entered. They release growth factors that signal to other cells, "We need repair here. Come help." Fibroblast cells respond, moving into the wound area. They begin producing collagen, the structural protein that gives skin its strength. The collagen forms a scaffolding, a framework for the new tissue. At the edges of the wound, skin cells begin dividing and migrating inward, slowly covering the exposed area. They're crawling over the collagen scaffolding, re-establishing the protective barrier. Blood vessels grow into the healing tissue, bringing nutrients and oxygen. Nerves begin reconnecting, restoring sensation. Within roughly 24 hours, the wound has regained significant strength and flexibility. It's not fully healed, that takes days or weeks depending on the size of the injury, but it's functional. The skin can stretch and move without breaking open again. Eventually, the scab falls away, revealing new skin underneath. It might be slightly discolored, slightly different in texture. There might be a scar if the wound was deep. But your skin has healed. It has closed the breach. It has maintained its function as a barrier between your interior and the world. The interior revolution. But skin is just the beginning. Inside your body, even more dramatic renewal is occurring. Your stomach lining, constantly exposed to acid strong enough to dissolve metal, is replaced entirely every few days. Think about that. The tissue lining your stomach is being destroyed and rebuilt continuously. The cells are sloughed off into the digestive content and new ones are generated to replace them, maintaining the protective barrier even as the old one is sacrificed. This is necessary because stomach acid doesn't discriminate. It breaks down food, but it would happily break down your stomach itself if not for this mucus layer and the constant regeneration of the cells producing it. Your stomach is, in essence, digesting itself slowly while simultaneously rebuilding. The renewal keeps pace with the destruction, maintaining the organ's integrity through continuous replacement rather than through ability. The battlefield of the gut. Your intestines face a different challenge. They're exposed to everything you eat and drink, including potential pathogens, toxins, and abrasive materials. The intestinal lining is continuously repairing damage from this constant assault. Cells are shed and replaced. Small wounds are sealed. Inflammation is managed. The entire lining of your small intestine is replaced roughly every 5 days. Every cell in that extensive surface area, which, if flattened out, would cover about 250 square meters, roughly the size of a tennis court, is replaced in less than a week. You're not aware of this replacement. It happens beneath the threshold of consciousness, managed by cellular programs that have been refined over hundreds of millions of years. But it's one of the most extensive regeneration processes in your body. You are rebuilding a tennis court's worth of tissue every 5 days, continuously, throughout your entire life. The liver's secret. And then there's the liver, that remarkable organ that filters your blood, produces bile, stores nutrients, and performs hundreds of other functions essential to life. The liver has a capability that sets it apart from most other human organs. It can regenerate after massive damage. Remove up to 70% of a healthy liver, and the remaining tissue will regrow until the organ reaches its original size again. Not by cell division throughout the liver, but through the expansion and proliferation of the remaining hepatocytes, the liver's main cell type. This isn't scar tissue filling the gap. It's functional liver tissue, capable of performing all the complex metabolic tasks the liver is responsible for. The regenerated liver isn't quite identical to the original. The architecture might be slightly different, the pattern of blood vessels altered. But it works. It performs its essential functions. The body can survive and thrive with a regenerated liver. This is the closest thing to true organ regeneration that humans possess. And it suggests that under the right conditions, our bodies retain more regenerative capacity than we usually give them credit for. Nerves that find their way. We often think of nerve damage as permanent, and for the central nervous system, the brain and spinal cord, this is largely true. Damage there is difficult to repair, and regeneration is limited. But peripheral nerves, the nerves in your arms, legs, and torso that connect your body to your spinal cord, can regrow. If a peripheral nerve is cut, specialized cells called Schwann cells can guide regenerating nerve fibers back to their targets. The Schwann cells form a channel, a pathway that the growing nerve follows, like a thread through a maze. The process is slow. Nerve regeneration proceeds at about 1 millimeter per day, sometimes slower. And it's not always perfect. The nerve might reconnect incorrectly, leading to odd sensations or incomplete recovery of function. But it's regeneration. True regrowth of neural tissue, reconnect ion of circuits, restoration of the ability to sense and move. If you've ever had a limb fall asleep from pressure on a nerve and felt the pins and needles sensation as it recovers, you've experienced what nerve regeneration feels like. That tingling is nerves re-establishing their connections, fibers finding their way back to the tissues they innervate. The geometry of healing. Mammals have evolved an interesting trick for healing large wounds. We shrink them. If you have loose skin, and mammals typically do, more so than many other animals, wounds can close not just by new tissue growing to fill the gap, but by the wound edges being pulled together. Specialized cells called myofibroblasts contract, pulling the wound margins toward each other. The surrounding skin stretches slightly to accommodate this contraction. The wound becomes smaller without requiring as much new tissue growth. This is why wounds heal faster in areas with loose skin than in areas where skin is tight. Your back heals differently than your shin. Your abdomen heals differently than the top of your foot. The geometry matters. The mechanical properties of the tissue influence how repair proceeds. The mystery of variation. Here's something puzzling. Closely related mammals heal at different speeds. Mice and rats, for instance, are quite similar genetically, but they heal wounds at measurably different rates. Some strains of mice are notably faster healers than others, even though they're all the same species. Humans show variation too. Some people are fast healers, their wounds closing and strengthening quickly. Others heal more slowly, taking longer to recover from the same injuries. Some of this variation is environmental, nutrition, age, overall health all affect healing speed. But some appears to be genetic, encoded in subtle differences in how repair genes are regulated or expressed. This variation suggests that healing speed is not fixed, not an immutable constant. It can be modified, enhanced, potentially improved through genetic or pharmaceutical interventions. The fact that one mouse strain heals faster than another means there are genetic variants that enhance repair. If we understood those variants, we might be able to induce similar enhancements in healing impaired individuals. The paradox of mammals. So here's the paradox. Mammals are extremely good at repair but generally poor at regeneration. We can heal wounds efficiently. We can replace skin, gut lining, liver tissue. We can regrow peripheral nerves. We maintain ourselves through constant cellular turnover. But we can't regrow a lost limb. We can't regenerate heart muscle after a heart attack. We can't rebuild a severed spinal cord. Why? What is it about mammalian biology that enables sophisticated repair but prevents true regeneration? The cost of complexity. One theory is that our complexity works against us. Mammals are warm-blooded, which requires high metabolic rates. We have sophisticated immune systems that might interfere with regeneration by treating regenerating tissue as foreign. We have complex nervous systems where precise reconnect ion is crucial. Random nerve regrowth isn't helpful if the nerves connect to the wrong targets. Our cellular specialization is extreme. Our cells commit strongly to particular identities, muscle cell, nerve cell, skin cell, and don't easily de-differentiate back to stem cell-like states the way axolotl or salamander cells can. This commitment enables the sophisticated functions our bodies perform. Your neurons can process information at incredible speeds precisely because they're so specialized. But that specialization comes at the cost of flexibility. A cell that's fully committed to being a neuron has largely lost the ability to become something else. And if it can't become something else, it can't participate in regenerating complex structures that require multiple cell types organizing in precise patterns. The cancer connection. There's another darker possibility. Maybe mammals suppress regeneration to prevent cancer. Cancer occurs when cells escape the normal controls on growth and division. They multiply without restraint, forming tumors, invading other tissues. Regeneration requires cells to divide, to proliferate, to move. All of these activities, if not tightly controlled, could lead to cancer. Perhaps mammals, with our long lifespans and high metabolic rates, have evolved to prioritize cancer suppression over regenerative capacity. Better to heal with scars than to risk uncontrolled cell growth. Some evidence supports this. Animals with greater regenerative capacity often have different cancer resistance mechanisms than mammals. They might be less susceptible to cancer despite having cells that can de-differentiate and proliferate freely. The trade-off might be real. You can have easy regeneration or strong cancer protection, but having both might be difficult. The revolution you don't see. But let's not undersell what your body accomplishes. You are constantly rebuilding yourself. Skin cells every month. Gut lining every few days. Liver tissue as needed. Blood cells continuously. Bone tissue gradually, remodeling in response to stress and use. The atoms in your body are not the atoms that were in your body years ago. You've replaced them gradually, through metabolism, through cellular turnover, through the simple fact of being alive in a universe where nothing stays the same. You are not a thing that persists. You are a process that persists. A pattern of organization that maintains itself through constant material exchange with the environment. You eat food, breathe air, drink water, and these materials become you. Temporarily. Until they're replaced in turn by new materials from new meals, new breaths, new drinks. The you that exists in this moment is made of materials that were part of the environment days or weeks or months ago. And the materials that make up this moment's you will return to the environment in time. The quiet miracle. Your body performs billions of repair events every day. Small cellular damages fixed. DNA lesions corrected. Proteins replaced. Membranes patched. Wounds sealed. Most of these repairs you never notice. They happen beneath the threshold of consciousness, managed by cellular machinery that works tirelessly from your first moment until your last. You don't have to think about replacing your stomach lining. You don't have to consciously direct skin cell division. You don't have to manage liver regeneration or nerve regrowth. Your body simply does it. The systems are in place, refined by hundreds of millions of years of evolution, operating with a reliability that's both humbling and extraordinary. You cannot regrow a severed arm. But you are regrowing your skin, your gut, portions of your liver, peripheral nerves, and countless cellular structures continuously. You are always healing, always repairing, always maintaining yourself against the universe's tendency toward disorder. Right now, in this moment, dead skin cells are falling from your body. New ones are rising to replace them. Your stomach lining is being rebuilt. Your intestines are regenerating. Somewhere in your body, a small wound is closing, collagen is being laid down, cells are migrating to restore barriers. You are being maintained. Continuously. Tirelessly. Quietly. This is not regeneration like an axolotl. This is not perfect repair like a zebrafish heart. But it's repair nonetheless. Aggressive, constant, effective enough to keep you alive and functional across decades. Your body cannot erase injury completely, but it can seal it, bridge it, work around it. It can maintain function despite imperfection. And perhaps that's enough. Perhaps the miracle isn't perfect regeneration. Perhaps the miracle is that repair works at all, that your body can take damage and respond, that wounds close, that life continues despite injury. You are scarred. You are imperfect. You are repaired rather than regenerated. And you are here. Still here. Maintained by systems that never stop, never rest, never give up on keeping you whole. Breathe slowly. Feel the quiet revolution happening within you. Cells dividing. Tissue regenerating. Damage being addressed, constantly, at every scale. You are not the same you from years ago. You've been rebuilt, gradually, piece by piece, through continuous repair. And you remain yourself. The pattern persists. The organization continues. This is your miracle. Not perfect, but real. And wonderfully, persistently, live. The wisdom of imperfection. Why we don't regenerate Eve. Writhing. There's a question that haunts regeneration research, whispered in laboratories and pondered in journal articles. If axolotls can regrow limbs perfectly, if zebrafish can regenerate hearts, if salamanders can rebuild spinal cords, why can't we? We share vast amounts of genetic similarity with these regenerators. We're all vertebrates. We all have the same basic toolkit of genes, the same fundamental cellular machinery. And yet, where they regenerate, we scar. Is this a failure of evolution? Did something go wrong in our mammalian lineage, some crucial ability lost? Or, and this is the more unsettling possibility, is scarring instead of regenerating actually adaptive? Are we imperfect on purpose? Tonight, we're going to explore one of biology's most profound paradoxes, that survival doesn't require perfection, and that sometimes limitations are features rather and bugs. The spectrum of repair. Evolution hasn't divided organisms into regenerators and non-regenerators, into healers and scarers. That would be too simple. Instead, regenerative capacity exists on a spectrum, a continuum of abilities that varies wildly even among closely related species. Some salamanders can regenerate limbs, tails, jaws, portions of their hearts, even parts of their brains and spinal cords. Others in the same family have more limited abilities. They can regrow tails but not limbs, or limbs but not complex organs. Among fish, zebrafish regenerate heart tissue beautifully, but many other fish species cannot. Among mammals, African spiny mice regenerate skin with remarkable fidelity, while most other rodents form prominent scars. There's no clear rule. No obvious dividing line. Just variation, endless, puzzling variation in regenerative capacity. This variation tells us something important. Regeneration is not a singular trait that you either have or don't have. It's a collection of capabilities, and evolution tinkers with them independently, enhancing some in certain lineages while reducing others. The heart of the matter. Consider hearts, those organs we cannot afford to lose, whose failure means death within minutes. Some vertebrates regenerate damaged heart tissue. Zebrafish, as we discussed, can rebuild substantial portions of their hearts. So can some newts and salamanders. But mammals, almost universally, cannot. When mammalian heart muscle dies, it's replaced with scar tissue. The heart becomes less elastic, less efficient. The scar doesn't contract like cardiac muscle does. This seems like a devastating limitation. Heart disease is among the leading causes of death in humans. How many lives could be saved if we could regenerate cardiac tissue? And yet, evolution hasn't given us this ability. Not because the genes don't exist. We have developmental genes that build hearts in embryos. But because those regenerative programs are shut down in adult mammals, inaccessible after we're born. Why? The trade-off hypothesis. One compelling answer is that evolution has traded away regeneration for other capabilities that, for mammals, proved more valuable. Warm-bloodedness requires high metabolic rates. Rapid growth allows mammals to reach reproductive maturity quickly. Complex nervous systems enable sophisticated behavior and learning. Strong immune responses protect against the pathogens that thrive at mammalian body temperatures. Each of these might be incompatible, in subtle ways, with maintaining easy regeneration. A high metabolic rate means cells are under more oxidative stress, accumulating damage faster, potentially making the dedifferentiation required for regeneration more risky. Rapid growth might require cells to commit strongly to specific identities, losing the flexibility needed to change into other cell types during regeneration. Complex nervous systems need extremely precise connections. Random regrowth of neurons could create harmful miswirings rather than restored function. Strong immune responses might attack regenerating tissue as foreign or aberrant, interfering with the healing process. The immortality problem. Here's another perspective that's both fascinating and slightly disturbing. If you could regenerate perfectly, indefinitely, you might never die. At least, not from injury or disease. You'd be vulnerable to predation, to starvation, to being crushed or burned beyond your repair capacity. But aging, organ failure, cancer, these might become surmountable with perfect regeneration. And immortality, or something approaching it, creates evolutionary problems. Evolution works through generations. Traits spread when organisms reproduce and pass on their genes. Beneficial mutations accumulate. Populations adapt to changing environments. But if individuals don't die and make room for the next generation, evolution slows dramatically. The species becomes less adaptable. When the environment changes, and it always changes, the species may lack the variation needed to survive. From the perspective of the species, not the individual, having members that age and die might be beneficial. It accelerates evolution. It ensures resources go to the young rather than being monopolized by the old. Perhaps limited regeneration isn't a failure. Perhaps it's a feature, a way of ensuring that individuals eventually exit the population, making room for the variation and adaptation that keeps the species viable across deep T. The longevity paradox. But the story gets more complex when we look at lifespan. Some animals live extraordinarily long lives: bowhead whales that may live over 200 years, Greenland sharks that might reach 400 years, certain clams that have been dated to over 500 years old. These long-lived animals don't regenerate better than their short-lived relatives. In fact, many regenerate worse. But they do have something else: exceptionally robust DNA repair mechanisms. Long-lived mammals upregulate many repair pathways compared to short-lived ones. They have more effective systems for detecting and fixing DNA damage, for managing oxidative stress, for clearing damaged proteins and dysfunctional cellular components. Better repair, at the molecular and cellular level, correlates strongly with longevity. Not better regeneration of organs or tissues, but better maintenance of the cells and molecules that make up those organs and tissues. It's as if evolution has two strategies for dealing with damage: fix small problems continuously, the long-lived strategy, or rebuild large structures occasionally, the regeneration strategy. And for mammals, the first strategy won out. Selective enhancement. But not all repair pathways scale equally with lifespan. Long-lived mammals don't just have uniformly better repair across the board. They have selectively enhanced certain pathways while leaving others at more modest levels. DNA repair is typically excellent in long-lived species. They cannot afford accumulated genetic damage over centuries. Protein quality control is also generally enhanced. Misfolded proteins need to be cleared before they cause problems. But other repair pathways aren't necessarily enhanced proportionally. Some antioxidant systems are better, but not dramatically so. Some inflammation responses are actually dampened rather than enhanced in long-lived species. This suggests that evolution is optimizing for something specific, not just more repair is better. There's a target, a balance point, where repair is good enough to enable the desired lifespan but not so aggressive that it creates other problems. The accuracy trade-off. Here's where things get really interesting. Some accurate repair pathways are deliberately down-regulated in certain circumstances. Take DNA mismatch repair, a system that fixes errors made during DNA replication. You'd think you'd want this running at maximum capacity all the time, right? Maximum accuracy, minimum errors. But actually, in some contexts, organisms reduce mismatch repair activity. During immune system development, for instance, deliberate mismatches create antibody diversity. During evolution of fast reproducing microbes, some level of mutation is beneficial for adapting to changing environments. Perfect accuracy isn't always optimal. Sometimes, a little sloppiness creates valuable variation. And very accurate repair has costs. The enzymes involved require energy. The process takes time. Cells with hyper-accurate repair systems grow more slowly than those with more relaxed standards. For organisms in competitive environments where rapid reproduction matters more than long-term survival, fast and sloppy might beat slow and accurate. The side effect problem. Every repair system has potential side effects. Inflammation helps clear damaged tissue and recruits repair cells, but chronic inflammation damages healthy tissue and accelerates aging. Scarring seals wounds quickly, but excessive scarring creates fibrosis that impairs organ function. Cell turnover replaces damaged cells, but each cell division carries a small risk of errors that could lead to cancer. Repair systems exist in a space of trade-offs. You're always balancing the benefit of fixing damage against the risk of the repair process itself causing problems. Evolution has calibrated these systems not for perfection but for optimal outcomes given the constraints. The amount of inflammation mammals experience after injury isn't randomly chosen. It's been tuned over millions of years to provide enough immune response to fight infection and clear debris without causing excessive collateral damage. The same is true for scarring, cell turnover, DNA repair, and every other repair mechanism. Each is set to a level that, on average, in the environments our ancestors faced, led to the best survival and reproductive outcomes. Not perfect outcomes. Best outcomes given the trade-offs. The cancer constraint. Let's return to cancer, because it may be one of the most important constraints on mammalian regeneration. Regeneration requires cell division. Lots of it. And cell division is inherently risky. Every time DNA is copied, there's a chance of errors. Most are caught and fixed, but some slip through. Mammals live long lives compared to most animals. Decades rather than months or years. This gives cancer many opportunities to arise. Perhaps mammals have evolved to suppress regeneration because the cancer risk over a long lifespan would be too high otherwise. The cellular programs that enable regeneration, dedifferentiation, proliferation, migration, are similar to the programs that cancer cells exploit. By keeping those programs shut down in adults, mammals may be protecting themselves from cancer at the cost of losing regenerative ability. It's a trade. Live longer without cancer, but don't regenerate when injured. For a mammal with a potential 70-year lifespan, that might be the better bet. For a salamander with a 10-year lifespan, the calculus is different. The optimization target. So what is evolution optimizing for? Not longevity alone. If it were, we'd all live like bowhead whales. Not regeneration alone. If it were, we'd all regenerate like axolotls. Not perfection in any single dimension. Evolution optimizes for reproductive success in specific environments. The organism that leaves the most viable offspring in its particular ecological niche is the one whose traits spread. For mammals in the niches we've occupied, the optimal strategy has apparently been repair molecular and cellular damage well enough to live for decades, heal wounds quickly even if not perfectly, grow rapidly to reproductive maturity, invest heavily in offspring care, accept the trade-offs of limited regeneration, aging, and eventual death. For salamanders in their niches, a different balance emerged: regenerate reliably, live more slowly, accept lower metabolic rates and longer maturation times, survive injuries that would be lethal to non-regenerators. Neither is better. They're solutions to different problems. The beautiful imperfection. There's something almost poetic about this realization. You are imperfect by design. Your inability to regenerate a limb isn't a cosmic mistake. It's the result of millions of years of evolutionary optimization that prioritized other traits that allowed your ancestors to survive, reproduce, and pass their genes forward. Your scars are part of a strategy that, on average, works better than perfect regeneration would in the context of mammalian life. You age and eventually die not because evolution failed to find a solution to aging, but because limited lifespan may actually benefit the species' long-term survival through accelerating generational turnover and adaptation. Your repair systems are calibrated to balance accuracy against speed, thoroughness against cost, healing against side effects, not randomly, but through a long process of trial and error spanning hundreds of millions of years. The spectrum we inhabit. Think of all the organisms we've discussed as points on a vast multidimensional spectrum of repair strategies. At one end, bacteria that reproduce so quickly that repair barely matters, just make new copies. At another, bowhead whales that repair so well they can live for centuries. In another direction, planarians that regenerate so completely they're nearly immortal. In yet another, mayflies that live for a day and invest nothing in long-term repair. Humans sit somewhere in the middle of this vast space. We're not the best at any single strategy, but we've combined moderate repair, moderate longevity, high metabolic rates, complex behavior, and parental investment in a way that's been extraordinarily successful by evolutionary standards. We've spread across the planet, adapted to every environment, built civilizations, created art and science and technology, all with bodies that scar instead of regenerate, that age instead of renewing indefinitely, that are imperfect in countless ways. Perhaps our imperfection is part of what made us successful. Perhaps limitations force creativity. Perhaps the fact that we can't regenerate lost limbs made us develop tools instead. Perhaps the fact that we age made us develop culture and knowledge transfer across genera. Tie-ins. You cannot regenerate a severed limb. You will age. Your DNA repair, while good, isn't perfect. Your wounds heal with scars. Your organ systems gradually decline. These aren't failures. They're features of a particular evolutionary strategy that has proven remarkably successful. Evolution has optimized you for survival and reproduction in the environments your ancestors inhabited. The trade-offs it made, scarring instead of regeneration, aging instead of indefinite renewal, moderate lifespan instead of centuries. You are alive. Your lineage has survived for billions of years, through countless environmental changes, through extinctions that eliminated most other species, through challenges we can barely imagine. Your imperfect repair systems are part of why you're here. Breathe slowly. Your scars, your limits, your mortality, these are not mistakes. They're part of a solution that works. Not the only solution. Not the best possible solution. But a solution that's carried your lineage from the first cells to you, across unimaginable spans of time. And that's enough. That's always been enough. You are a point on evolution's spectrum of repair strategies. Imperfect, limited, mortal, and wonderfully, persistently alive. Survive first, perfect never. The pragmatism of plants and microbes. Imagine you're in a drought. Water is scarce. Every drop matters. And then someone cuts you, a deep wound that exposes your interior to the dry air. You don't have time to rebuild perfectly. You don't have resources to waste on aesthetics. You need to seal that wound immediately, before you lose the water that's keeping you alive. This is the daily reality for plants. While we've been exploring regeneration in animals, the complexity of cellular repair, the marvel of regrown limbs, the mystery of scarless healing, plants have been solving the same fundamental problem with a completely different set of constraints. Plants can't run from danger, can't seek shelter, can't migrate to better conditions. They're rooted in place, literally, vulnerable to whatever comes. Browsing animals, cutting winds, falling branches, human pruning shears. And so they've evolved repair strategies that prioritize one thing above all else: survival. Not beauty. Not perfection. Not complete restoration. Just staying alive long enough to reproduce. The seal, not the cure. When a plant is wounded, when a branch breaks, when an animal tears leaves, when you prune a stem, the first response is immediate and urgent. Seal the wound. Not heal it. Not regenerate it. Just seal it. Plants lose water through their surfaces. In intact plants, this is controlled by specialized pores called stomata that can open and close. But a wound is an uncontrolled opening, and through it, water escapes freely into the air. For a plant, especially in dry conditions, this water loss can be fatal. Dehydration kills faster than infection, faster than nutrient loss, faster than almost any other consequence of injury. So the plant's first priority is plugging the leak. The chemistry of closure. Many plants accomplish this through rapid chemical changes at the wound site. Damaged cells release compounds that oxidize when exposed to air, forming barriers that reduce water loss. Phenolic compounds crosslink, creating a temporary seal. Specialized cells near the wound site begin producing suberin or lignin, complex, waxy polymers that are impermeable to water. This isn't healing in the sense we usually mean. The tissue isn't being repaired. Cells aren't regenerating. The plant is essentially applying a molecular band-aid, a chemical seal that stops the immediate crisis. And it happens fast. Within hours in many cases, the wound is sealed well enough to prevent catastrophic water loss. Only after this emergency seal is in place does the slower process of actual tissue repair begin. The callus solution. Once the wound is sealed, plants begin forming callus tissue, a mass of undifferentiated cells that fills the gap left by injury. Callus tissue is plant scar tissue, essentially. It's not specialized. It doesn't have the structure of normal plant tissue, no organized vascular bundles, no specialized cell types performing specific functions. It's just cells, filling space, maintaining structural integrity. Over time, weeks, months, sometimes years, this callus tissue may gradually differentiate into more organized structures. Vascular tissue might form to reconnect severed transport pathways. Strengthening fibers might develop to provide mechanical support. But this is slow. Plants don't prioritize rebuilding. They prioritize surviving, and if survival is possible with a mass of callus tissue rather than perfectly regenerated structure, that's acceptable. The reproduction priority. Here's what's fascinating. Plants actually deprioritize growth and repair in favor of reproduction when resources are limited. If a plant is damaged and resources are scarce, it often reduces investment in regrowing lost structures and instead channels available resources into producing flowers and seeds. It's as if the plant calculates, I may not survive long. I may not be able to rebuild completely. So rather than invest everything in trying to restore what I've lost, I'll invest in leaving offspring. Height doesn't matter if you die before reproducing. A perfect crown of leaves doesn't matter if you can't make seeds. This is survival thinking at its most pragmatic. Repair what's necessary to stay alive, but don't obsess over perfection when the clock is ticking. The hydraulic engineers. Some plants use their internal hydraulic systems to aid wound closure in remarkably sophisticated ways. Cacti, for instance, operate under substantial internal pressure. Their tissues are turgid, pressurized by water. When wounded, this pressure can actually help close the wound mechanically. The pressurized tissue around the wound site swells slightly, pushing wound edges together. This mechanical closure supplements the chemical sealing, providing an additional barrier against water loss. The cactus doesn't need to actively do anything. The pressure already present in its tissues, pressure that normally maintains the cactus's structure and rigidity, becomes part of the wound response. It's elegant engineering, using existing properties of the system to solve new problems without requiring additional specialized mechanisms. Liquid bandages. Some trees have evolved an even more dramatic immediate response. They bleed. Not blood, of course, but latex, thick, milky fluids that flow out of wounds in species like rubber trees, milkweeds, and figs. This latex serves multiple functions. It's toxic or unpalatable to many herbivores, discouraging further damage. It physically blocks the wound, preventing water loss and pathogen entry. And crucially, it coagulates. Like blood clotting in animals, plant latex undergoes chemical changes when exposed to air that transform it from liquid to solid. The fluid that flows out becomes a plug that seals the wound. This coagulation depends on the internal pressure of the plant's vascular system. The pressure forces latex out of the wound initially, then the coagulation traps that latex in place, forming a seal. Too little pressure, and not enough latex emerges to seal the wound effectively. Too much pressure, and the plant loses excessive fluid before coagulation can occur. The system is calibrated, balanced through evolution to provide optimal wound sealing given the constraints of plant physiology. Time and patience. After the immediate sealing, plant repair proceeds slowly. Where animals might close a wound in days and rebuild tissue in weeks, plants take months or years. A tree wounded in spring might not fully seal that wound until the following growing season, or the one after that. This isn't inefficiency. It's a different strategy based on different constraints. Plants photosynthesize. They create their own energy from sunlight. They don't need to maintain high metabolic rates to hunt for food or escape predators. They can afford to be slow. And being slow has advantages. It's energetically cheaper, it produces less risk of mistakes, it allows the plant to allocate resources flexibly depending on current conditions. If this year brings good conditions, ample rain, plenty of light, the plant can invest more in repair. If conditions are harsh, it can reduce that investment and focus on basic survival instead. This flexibility, paradoxically, comes from not being in a hurry. The microbial fortress. Now let's shift scale dramatically, from trees down to bacteria. Bacteria face threats that would seem apocalyptic at our scale. Antibiotics that destroy cell walls. Immune cells that engulf and digest them. Toxic chemicals in their environment. Desiccation. Starvation. Viral infections. A single bacterium, alone, is fragile and vulnerable. But bacteria don't typically face these challenges alone. They form communities, biofilms, where thousands or millions of bacteria live together in a self-produced matrix of polymers and proteins. These biofilms are bacterial cities, and they're remarkably resistant to damage. The shield of slime. The matrix surrounding bacteria in a biofilm acts as a physical barrier. Antibiotics diffuse through it slowly, giving bacteria time to produce enzymes that break down the drugs before they reach lethal concentrations. Immune cells have difficulty penetrating it. Environmental toxins are diluted or neutralized. But the biofilm is more than just armor. It's a coordinated community where bacteria communicate with each other through chemical signals, where some bacteria specialize in producing the protective matrix while others focus on nutrient acquisition, where damage to one part of the biofilm triggers responses elsewhere. When part of a biofilm is destroyed, by mechanical disruption, by immune attack, by antibiotic treatment, surviving bacteria detect the damage and respond. They increase production of matrix components, thickening the protective layer. They alter their metabolism to survive in a damaged, stressed environment. They may release factors that help other bacteria in the biofilm survive. And if the damage is severe enough, bacteria at the edges of the intact biofilm can disperse, leaving to colonize new sites, ensuring that even if this particular biofilm is destroyed, the bacterial lineage survives elsewhere. Strength in diversity. Here's something remarkable. Microbial communities containing multiple species repair damage more effectively than single-species biofilms. Different bacterial species bring different capabilities. Some produce especially robust matrix materials. Others generate nutrients that feed the community. Still others produce antimicrobial compounds that protect against competitors. When damage occurs, this diversity becomes crucial. If one species is particularly vulnerable to whatever caused the damage, others may be more resistant. The community can lose members and still function. It's resilience through redundancy. Multiple species providing overlapping functions means no single point of failure. This parallels patterns we see in ecosystems. Diverse forests recover from disturbance more reliably than monocultures. The principle scales from microbial films to entire biomes. Diversity confers resili. NCE. Survival over elegance. What plants and microbes teach us is that repair doesn't have to be elegant to be effective. The tree doesn't need to perfectly regenerate lost branches. It needs to seal wounds, prevent water loss, and survive to the next growing season. The cactus doesn't need scarless healing. It needs to close wounds quickly using internal pressure, preventing dehydration in desert conditions. The bacterial biofilm doesn't need to maintain perfect structure. It needs to resist antibiotics long enough for the colony to survive and disperse. These organisms have evolved repair strategies that prioritize staying alive over looking untouched. And those strategies work. They've kept these lineages alive for hundreds of millions of years. The honesty of scars. There's something almost honest about plant wounds. The tree doesn't hide its scars. The healed-over wound remains visible, sometimes for decades, as a different texture of bark, a slight depression in the trunk, a deviation in the growth pattern. These scars tell the tree's history. The branch lost in a storm 20 years ago. The deer browse from last winter. The pruning cut you made when the tree was young. The tree carries its past in its form. It doesn't erase damage, it incorporates it, grows around it, makes it part of the structure. In a sense, the scarred tree is more interesting than an undamaged one. Its form reflects its experiences, its survival through challenges, the specific path it took through. Time. The collective response. Bacteria in biofilms demonstrate something that animals rarely achieve: truly collective repair. When part of a biofilm is damaged, the response isn't individual. Bacteria throughout the community alter their behavior. Some increase matrix production. Others change their metabolism. Some disperse to establish new colonies. The biofilm repairs as a collective, with specialized roles and coordinated responses that no single bacterium could accomplish alone. This is repair as an emergent property of community organization. No central control. No master plan. Just local responses that, when aggregated across thousands of bacteria, produce community-level healing. It's reminiscent of ant colonies repairing their nests, or bee swarms replacing lost members. The intelligence is distributed, the response collective, the outcome greater than what any individual can achieve. Pragmatism as strategy. What all these examples share is pragmatism. The plant doesn't try to achieve perfect regeneration. It seals wounds quickly and rebuilds slowly, prioritizing reproduction over complete restoration. The cactus uses what it already has, internal pressure, to aid wound closure rather than evolving entirely new mechanisms. The tree-bleeding latex turns an anti-herbivore defense into a wound sealing system, one adaptation serving multiple functions. The biofilm doesn't try to be invulnerable, it accepts that damage will occur and builds in redundancy and dispersal capabilities to survive it. These are solutions that work within constraints, that make the best of limited resources, that prioritize effectiveness over perfection. The beauty of good enough. There's a concept in engineering called satisficing, choosing solutions that are satisfactory and sufficient rather than optimal. Good enough rather than perfect. Plants and microbes are masters of satisficing. Their repair strategies aren't the best possible, they're the best achievable given their constraints and the trade-offs they face. The tree could potentially invest more resources in wound healing, achieving faster, more complete repair. But those resources would come from somewhere, from growth, from reproduction, from storing reserves for winter. The trade-off wouldn't be worth it. The biofilm could potentially produce thicker protective matrices, more robust structures, better resistance to antibiotics. But thicker matrices require more resources to produce, take longer to build, might be less flexible in responding to environmental changes. Good enough wins. Perfect loses. Survival trumps aesthetics. In forests and fields, in deserts and wetlands, in the invisible microbial films coating every surface, repair is happening constantly. Not elegant repair. Not perfect repair. Just effective repair, good enough to prevent death, sufficient to enable reproduction, adequate to keep the lineage going. Trees are sealing wounds with coagulating latex. Cacti are closing breaches hydraulically. Callus tissue is slowly filling gaps in damaged stems. Bacterial communities are thickening their protective matrices and dispersing to establish new colonies. None of them are trying to be perfect. They're trying to survive. And they do. They persist through droughts and storms, through herbivore attacks and pathogen invasions, through all the countless insults that living in a physical world entails. Their scars tell stories of survival. Their imperfect repairs are badges of resilience. You, too, carry scars, physical and metaphorical. Places where you were damaged and healed imperfectly. Wounds that sealed but didn't erase. Perhaps those scars are not failures but evidence of pragmatism, of the wisdom of good enough, of prioritizing survival over perfection. Perhaps, like the scarred tree that continues growing, like the biofilm that repairs collectively, you are most yourself not in your perfection but in your resilient imperfection. Breathe slowly. Feel yourself as part of this vast community of repairing life. Plants sealing wounds, microbes rebuilding communities, all of us surviving first, perfecting never. Good enough is good enough. It always has been. It's what's kept life going for billions of years. And it will keep you going too. Learning from life. When materials heal themselves. Yes. For most of human history, we've built things to resist damage. We've made materials harder, stronger, more rigid. We've designed structures to withstand stress, to bear loads, to endure forces without breaking. When something broke, we replaced it or patched it from the outside. We've been fighting damage, trying to prevent it, trying to make things that never fail. But nature, as we've seen tonight, takes a completely different approach. Nature accepts that damage will happen. It builds systems that sense damage automatically and repair it autonomously. It creates materials that heal themselves, structures that rebuild, organisms that regenerate. And now, finally, human engineers are learning from this wisdom. We're beginning to create materials that don't just resist damage, they heal from it. We're building systems inspired by the self-repair we see in cells, in tissues, in organisms across the tree of life. We're moving from fighting damage to accepting it, and in that acceptance, finding new possibility. Is the inspiration. Think about everything we've explored tonight. Cells that sense mechanical stress through force-sensitive proteins and rush repair molecules to damaged sites. Membranes that seal breaches within seconds using calcium-triggered vesicle fusion. Organisms that regrow entire body parts. Tissues that regenerate without scarring. Each of these represents a solution to the universal problem of damage, a solution refined over hundreds of millions of years, tested in every imaginable environment, proven to work across billions of organisms. These solutions are blueprints, waiting to be translated into human technology. And that translation is happening now, in laboratories around the world, where engineers and biologists are working together to create materials that behave like living tissue, sensing damage, responding autonomously, repairing themselves without external intervention. Tyon. The soft revolution. One of the most promising applications is in medicine, where materials need to interface with the soft, dynamic tissues of the human body. Consider vocal cords. They vibrate hundreds of times per second when you speak or sing. They need to be flexible, resilient, able to endure millions of cycles of stress and relaxation. When vocal cords are damaged, by overuse, by surgery, by disease, repairing them is challenging. Hard materials don't flex properly. Stiff patches restrict movement. Traditional repair methods often leave people with permanently altered voices. But now, researchers have developed self-healing hydrogels that match the softness of vocal cord tissue. These hydrogels are mostly water, held together by polymer networks. And crucially, when damaged, they can heal autonomously. Cut the hydrogel, and the severed polymer chains find each other again. They rebond. They restore the material's integrity. Within hours, the hydrogel has recovered most of its original strength and flexibility. This happens without external intervention. No surgeon repairing it. No glue applied. The material simply heals itself, the way tissue does. Moving joints. Think about your knees, your elbows, your shoulders. Joints that move constantly, that bear enormous forces, that experience friction and stress with every step, every reach, every motion. When cartilage in these joints degenerates, the result is pain, inflammation, limited mobility. Traditional treatments include injecting lubricants or, in severe cases, joint replacement surgery. But injectable hydrogels offer a different approach. These materials can be injected directly into joints, where they flow into damaged areas and then solidify. And because they're self-healing, they continue functioning even as the joint moves and stresses them. The hydrogel experiences microdamage constantly as you walk, bend, reach. But it repairs that damage continuously, maintaining its protective and lubricating functions without degrading rapidly. It's inspired by how cartilage itself works. Cartilage is constantly experiencing microdamage and micro-repair, maintaining itself through a balance of breakdown and restoration. The synthetic hydrogel mimics this, creating a material that accepts damage as part of its normal function and heals as part of its normal behavior. Building bone. Some hydrogels go even further. They don't just heal themselves, they help the body heal. Researchers have developed hydrogels that, when implanted in bone defects, gradually break down while supporting new bone growth. The hydrogel acts as a scaffold, giving bone cells something to attach to and grow on. And if the hydrogel is damaged during this process, by movement, by mechanical stress, by the activity of cells growing through it, it heals itself, maintaining its structural integrity long enough for natural bone to replace it. The same principle applies to tooth repair. Dentin, the hard tissue under tooth enamel, doesn't regenerate well in adults. But hydrogels that can form in the complex, confined spaces inside teeth, that can withstand the forces of chewing while healing their own damage, offer new possibilities for preserving teeth that would otherwise need to be extracted. These materials are temporary by design. They heal themselves long enough to do their job, then gradually break down as natural tissue replaces them. It's not permanent repair. It's scaffolded regeneration, with the scaffold itself capable of self-maintenance. The chemistry of reconnection. But how do these materials actually heal themselves? Some use a principle inspired by how polymers behave naturally. Entanglement. Imagine two bundles of yarn. If you cut through both bundles and then press the cut ends together, the individual strands can interweave, can find each other across the cut, can re-entangle. Some synthetic polymers do exactly this. When cut, the long molecular chains at the severed surfaces can migrate across the interface, can intertwine with chains from the other side, can reform a continuous network. No chemical bonds are being formed, or rather, only weak, reversible bonds. But these weak bonds, multiplied across millions of molecular interactions, are enough to restore the material's strength. The polymer remembers, in a sense, how it's supposed to be organized. And when damaged, it spontaneously returns to that organizatio. N. Liquid armor. Other materials take a completely different approach, inspired by how some biological fluids behave under stress. These materials are called shear-thickening fluids. Under normal conditions, they flow like liquids. But when subjected to sudden impact, when stressed rapidly, they solidify temporarily. It's like the non-Newtonian fluids you might have played with as a child, mixtures of cornstarch and water that you can run across but sink into if you stand still. Engineers have developed materials that use this principle for protection. Flexible armor that's soft and comfortable until struck, then momentarily becomes rigid to absorb impact, then returns to flexibility. The material isn't healing in the conventional sense, it's never broken. Instead, it's continuously adapting its properties to the forces being applied, becoming hard when it needs to be hard, soft when it needs to be soft. It's damage prevention through dynamic response, inspired by how biological tissues adapt their properties. Muscle that can be relaxed or tense, skin that can be supple or taut. Combining strategies. Some of the most sophisticated self-healing materials use hybrid approaches, combining multiple repair mechanisms the way organisms do. Intrinsic healing mechanisms are built into the material itself. Polymers that can re-entangle, chemical groups that can rebond spontaneously. Extrinsic healing mechanisms involve embedded capsules of healing agents, like the calcium-loaded vesicles in cells that rush to membrane wounds. When the material is damaged and these capsules rupture, they release chemicals that polymerize or cross-link, filling the crack. A material might use both, Intrinsic healing for small damages that occur constantly, extrinsic healing for large damages that intrinsic mechanisms can't fully address. It's a two-tier system, analogous to how organisms have both continuous cellular repair and dramatic wound healing responses depending on the scale of damage. Living materials. And then there are materials that literally are alive, at least partly. Mycelium, the network of fungal threads that makes up most of a fungus's body, can be grown into specific shapes and then killed and dried, producing a material that's lightweight, strong, biodegradable. But some researchers are working with living mycelium materials, structures where the fungus is kept alive and continues growing. These materials can regenerate internal damage. If the mycelial network is disrupted, the fungus grows new threads, reconnecting severed pathways, restoring the material's structural integrity. Some versions even incorporate fungal spores as repair seeds, dormant structures embedded throughout the material that activate when damage occurs, germinating and growing to fill gaps. It's not a synthetic material mimicking biology. It's actual biology, co-opted and shaped to serve human purposes while retaining its living properties. The acceptance revolution. What all these materials represent is a fundamental shift in engineering philosophy. For centuries, we've designed for perfection. Materials that don't fail, structures that don't break, systems that don't degrade. But this pursuit of perfection has limits. Materials become brittle when too strong. Systems become fragile when too optimized. The attempt to prevent all damage often makes catastrophic failure more likely when damage does inevitably occur. Nature has never pursued this kind of perfection. Nature accepts that damage will happen and builds systems to deal with it. And now, engineering is learning the same lesson. Self-healing materials accept that damage occurs. They're designed with repair mechanisms already built in, waiting to activate when needed. This makes them more resilient, not despite accepting damage but because they accept it. They can survive conditions that would destroy traditional materials, not by preventing damage but by continuously healing it. The cost question. Of course, self-healing comes with costs, just as it does in biology. These materials are often more complex to manufacture. The healing mechanisms add weight or reduce other performance characteristics. The repair process takes time, during which the material may be weakened. And there's energy involved, whether it's the chemical energy stored in healing capsules, or the metabolic energy required to keep mycelium alive, or the heat or light needed to trigger some healing reactions. Engineers face the same trade-offs nature does. How much should you invest in repair capability versus other properties? How quickly does healing need to occur? What scale of damage should the material be able to address? The answers depend on the application. A biomedical implant needs different healing capabilities than a structural beam. A coating for spacecraft needs different repair mechanisms than a flexible electronics component. But the fundamental principle remains. Accepting damage and building in repair capacity is often better than trying to prevent all damage. Beyond mimicry. The most exciting possibility is that we might eventually create materials that exceed what nature has achieved. Biology is constrained by evolution, by what mutations can arise randomly, by what can develop from existing structures, by what's compatible with the organism's other needs. Human engineering doesn't face exactly the same constraints. We can combine mechanisms from different organisms. We can use synthetic chemistries that don't exist in nature. We can optimize for specific applications in ways evolution never could. Imagine materials that heal faster than any biological tissue, that sense damage more sensitively than any cell, that can repair themselves repeatedly without degrading, without accumulating the costs that biological repair imposes. These possibilities are no longer science fiction. They're active areas of research, with early versions already being tested. The philosophical shift. But perhaps more important than any specific material is the shift in thinking these developments represent. We're learning to see damage not as failure but as an expected part of a material's life cycle. We're designing systems that work with damage rather than fighting against it. We're recognizing that resilience, the ability to survive and recover from damage, is often more valuable than durability, the ability to resist damage in the first place. A bridge that can heal small cracks continuously might last longer than one built to never crack, but that fails catastrophically when cracks eventually appear. A medical implant that repairs its own damage might function better over decades than one that's initially perfect but gradually degrades without repair. This is the wisdom biology has embodied for billions of years, and we're just beginning to appreciate and apply it. The materials of tomorrow. In laboratories today, researchers are developing concrete that heals its own cracks using embedded bacteria that produce limestone when water enters the crack. Coatings for ships and aircraft that repair scratches autonomously, preventing corrosion. Flexible electronics that can be bent, folded, even cut, then heal themselves to restore function. Soft robots that repair damage to their pneumatic actuators, allowing them to continue operating even after punctures. Biodegradable packaging that heals tears and holes, extending shelf life while remaining compostable. Each of these represents a small step toward a future where our materials behave more like living tissue, self-maintaining, self-repairing, resilient rather than merely strong. We stand at a threshold. For most of human history, we've built static things, objects meant to resist change, to endure unchanging through time. But we're learning from biology that the key to longevity isn't resisting change, it's managing it. It's accepting damage and repairing it. It's building systems that heal themselves, that maintain their function through continuous self-correction rather than through unchanging perfection. The materials emerging from laboratories today are the first generation of a new kind of technology, one that works with the grain of physical reality rather than against it. They accept that damage will occur. They incorporate repair mechanisms. They prioritize resilience over rigidity. They're learning from life, from the billions of years of evolutionary refinement that have produced organisms that sense damage instantly, repair it autonomously, and persist through cycles of breakdown and restoration. You've been experiencing this all night, listening to stories of repair, of regeneration, of resilience in the face of damage. Your own body embodies these principles. You are self-healing material, maintained through continuous repair, persisting through constant renewal. And now, the materials we create are beginning to share these properties. We're building a world where damage doesn't mean the end, where breaking doesn't mean failure, where healing is built into the structure of things. It's a world inspired by life. A world that accepts imperfection and builds in repair. A world that's more resilient because it stops fighting the inevitable and instead learns to heal. Breathe slowly. Feel the continuity, from cells that patch membranes in seconds, to organisms that regrow limbs, to materials that heal their own cracks. The principle is the same. Damage is inevitable. Repair is possible. Resilience comes not from avoiding damage but from recovering from it. This is biology's gift to engineering. And perhaps, it's biology's gift to you as well, the knowledge that you don't have to be undamaged to be whole. You just have to be able to heal. The living future, when technology breathes. Imagine a building that heals its own cracks. Not with machines, not with human intervention, but with living cells embedded in its concrete, waking from dormancy when water enters a fissure, producing limestone to seal the breach, then returning to sleep until needed again. Imagine clothing that repairs its own tears, not with thread and needle, but with bacterial films that sense damage and grow new fibers to bridge the gap. Imagine metals that flow like liquid to fill their own fractures, then resolidify, recovering nearly all their original strength as if they were never broken. These aren't fantasies. They're not distant dreams. They're technologies being developed right now, in laboratories around the world, blurring the boundary between the living and the made, between biology and engineering, between organism and object. We stand at the threshold of a new era, one where the things we build don't just mimic life's repair strategies. They incorporate life itself. Clothing that lives. Your clothes are made of dead materials, cotton fibers from plants that were harvested months or years ago, synthetic polymers created in chemical reactors, dyes and treatments applied to inert fabrics. When your shirt tears, it stays torn until you mend it or discard it. But researchers are developing textiles embedded with living bacterial films, thin layers of microorganisms integrated into the fabric structure itself. These bacteria remain dormant most of the time, requiring minimal resources, barely affecting the textile's properties. But when the fabric is damaged, when fibers break, when tears appear, the bacteria sense the disruption. They activate. They begin producing polymers and proteins that bridge the gap, that connect severed fibers, that fill tears with new material. Over hours or days, the fabric repairs itself. The tear closes. The material regains its strength. The bacteria return to dormancy, waiting for the next damage event. Your shirt becomes a living thing, not alive in the way you are, but alive enough to sense damage and respond to it, to maintain itself rather than simply degrading passively over time. Buildings that heal themselves. Concrete is one of humanity's most widely used materials. We've built our civilization on it. Roads, bridges, buildings, dams. Trillions of tons of concrete shape the modern world. But concrete cracks. Water enters those cracks, corrodes the steel reinforcement inside, and the structure weakens. Eventually, catastrophically, it fails. Repairing concrete is expensive, labor-intensive, and often incomplete. We patch what we can see, but cracks internal to the structure go unaddressed. Now, imagine concrete that repairs itself from within. Bio-self-healing concrete contains bacteria, typically Bacillus species that can survive the harsh, alkaline environment inside concrete. These bacteria are embedded in the material along with calcium-rich nutrients, all encased in protective capsules. When cracks form and water enters, the capsules break open. The bacteria, dormant until this moment, activate. They consume the nutrients and produce calcium carbonate, limestone, as a metabolic byproduct. This limestone precipitates in the crack, gradually filling it, sealing it, preventing further water intrusion. The bacteria are doing exactly what they would do in nature, metabolizing nutrients, producing minerals. But here, in concrete, their natural behavior becomes structural repair. Engineering evolution. But the bacteria initially used weren't optimal for this application. They worked, but not ideally. They sometimes produced too little limestone, or the wrong crystal structure, or they died before the crack was fully sealed. So researchers turned to genetic engineering. They're modifying bacteria to be better masons, to produce more limestone, more quickly, in crystal forms that bond better with concrete. They're engineering bacteria that can survive longer in dormancy, that activate more reliably when exposed to water, that coordinate their limestone production more effectively. This is evolution accelerated and directed. Instead of waiting millions of years for natural selection to optimize bacteria for concrete repair, we're doing it deliberately, in years or decades. The bacteria are becoming not just living organisms but engineered biological components, designed, tested, refined for specific functions in human-built structures. Cold survival. One challenge is that concrete exists in all climates. A bridge in Norway experiences temperatures well below freezing for months each year. Can bacteria survive that? Can they still repair cracks in cold conditions? Some microbes, it turns out, remain metabolically active near freezing temperatures. These cycrotolerant bacteria are adapted to cold environments, Arctic soils, deep oceans, permanently cold caves. They've evolved enzymes that function at low temperatures, membranes that stay fluid when cold, metabolic pathways that proceed even when thermal energy is minimal. By incorporating these cold-adapted bacteria into concrete, or by engineering standard bacteria to express cold tolerance genes, researchers are creating concrete that can self-heal even in winter, even in climates where traditional concrete repair would be impossible for months at a time. The building doesn't care that it's cold. Its embedded repair system continues functioning, maintaining structural integrity year-round. As you drift toward sleep tonight, feel the repair happening within you. Skin cells dividing, rising, replacing what was shed. DNA being checked and corrected by vigilant enzymes. Proteins being folded and refolded, or tagged for replacement if damaged beyond repair. Membranes being patched. Tissues being maintained. You are being healed, constantly, at every scale. You are being rebuilt, continuously, through processes older than mountains. And somewhere in the world, a crack in concrete is being sealed by awakening bacteria. A textile tear is being bridged by bacterial polymers. Liquid metal is flowing to fill a fracture. An artificial muscle is sensing and repairing its own damage. The line between you and these materials grows thinner. The principle is the same. Accept damage. Sense it. Respond to it. Rebuild what was broken, or work around it if rebuilding isn't possible. Persist. Heal. Continue. This is the wisdom of 4 billion years of life. And it's enough. It's always been enough. You are imperfect. You are scarred. You are continuously damaged and continuously repaired. And you are here. Still here. Maintained. Renewed. Alive. Rest now. Let your repair systems do their quiet work. Trust the ancient machinery that has kept your lineage alive across unimaginable time. You will wake renewed, slightly rebuilt, never quite the same but always yourself. The pattern persists. The organization continues. The repair never ends. And that is beautiful. That is enough. Sleep well.